questions from last week? All right, great. If there are no questions, then let's begin with today's assignment. Okay. I believe you can all see the screen clearly. Okay. All right. So today I want to talk about the endocrine system. The endocrine system. Okay. So this is another important topic which I believe you should know very well, especially not for the test alone, but for your clinical practice as well. Because when you start your clinical practice, your nursing courses, you'll be talking about abnormalities of the endocrine system. They are in what we call pathophysiology. Okay, so it's good to know the normal, then you can work out the abnormal when you get there. Okay, so know the endocrine system very well. And for this test, as I said, most of the things are all like basic and general knowledge. You're not going to go too much into the details of each of them. Because if you take one system alone, one endocrine system, what do you call it? One part of the endocrine system alone, like the hypothalamus, we can spend days just talking about it. Or the pituitary, can spend several days talking about pituitary gland alone, and you never finish. Some people, their whole life is just to study the pituitary gland. For the test, just a general knowledge about of everything, general. Okay, and we're going to see how they ask a question on this after the lesson. So most of the time they'll give a diagram like I have here, and then the different parts will be labeled 1, 2, 3, 4, 5, and so on. And then they'll ask you questions based on that. Okay, so what's the function of the organ label? Maybe 1, or the one labeled 2, 3, and so on. Okay, as I said, we'll see questions like that shortly. So we're going to look at the different parts of the endocrine system, so you have a complete knowledge or overview of it. Okay, so when you say an endocrine gland refers to glands that don't have ducts. They don't have ducts, so we call them the ductless glands without ducts. So very important to note this here. Glands without ducts. Okay, so basically there are two types of glands. Those that have ducts, which we call exocrine. You have the exocrine gland. So let's put that here. Types of glands. You have exocrine and endocrine. Okay. So exocrine glands, when they have ducts, glands with ducts. Ducts are opening to the surface. So if you take the gland, it will be something like this, epithelial tissue in there, and they have like an opening. It comes to the surface, opens the surface through ducts. So the duct will be like the tube here, like these are the ducts to the surface. Okay. Like the salivary gland. If there's a salivary gland, the salivary gland is an exocrine gland. Okay. The endocrine gland, these have no ducts, as I've just said, no ducts. So we call them the ductless glands, ductless, no duct. So you just see a mass of tissue, maybe a mass of tissue like this. And then you have a lot of blood vessels that will go in and out. Blood vessels going in and out because they release their secretions directly into the blood. Everything goes directly into the blood. It's the blood vessels. Okay. So they give you a question like somebody's looking under the microscope and the person notices that the gland has a lot of blood vessels entering and leaving it. what type of gland is the person looking at, then you have to know that the person is looking at endocrine gland. Okay, because they release their secretions directly into the blood. Okay, good. So those are two types of gland. But today, as I said, we are looking at the endocrine gland. Okay. Now the endocrine glands, they produce what we call secretions. So the chemicals that they release, we call them secretions. So the secretions are released directly into the blood. The endocrine glands secrete hormones. Okay, they produce hormones, and the hormones, they regulate various metabolic activities in the body. Okay. So here's a diagram of the endocrine system, as I said. So let's go through the different types. Now, we'll start with the hypothalamus. Okay, so the hypothalamus somewhere here, the hypothalamus around here. Okay, and then the hypothalamus produces 8 hormones. It produces about 8 hormones. Okay? Some of the hormones, they stimulate the pituitary gland to produce other hormones. Okay, so it will be good to have an idea of the hormones released by the hypothalamus. Okay, one way of remembering them, you see the word releasing hormone attached to them. So if I go down here, I have the 8 different hormones released by the hypothalamus. Yeah. So one of them is called a thyrotropin releasing hormone, number 1. Okay, so this acts on the pituitary to release thyroid stimulating hormone, okay, which we call the TSH. We'll talk about that. And then it also releases growth hormone releasing hormone, okay, GHRH, growth hormone releasing hormone. So this acts on the pituitary to release growth hormone, which we call the GH. Then you have the gonadotropin releasing hormone, gonadotropin releasing hormone, GRH. This acts on the pituitary to release what we call the gonadotropins. And then the gonadotropins are the follicle stimulating hormones, FSH, and the luteinizing hormone, we call LH. And then you have corticotropin releasing hormone, CRH. This acts on the pituitary to release the adrenocorticotropic hormones, adrenocorticotropic hormones. Okay, we'll talk about the function later. Then it also releases oxytocin. Oxytocin, this causes uterine contractions and helps with the secretion of breast milk during lactation. So that's oxytocin. Then dopamine, it also produces dopamine. Dopamine is what we call the neuroendocrine transmitter. Okay, it acts on the neurotransmitter. It acts as neurotransmitter and also stimulates the release of other hormones. It stimulates the release of other hormones, dopamine. And then have vasopressin. Another name for vasopressin is the ADH, anti-diuretic hormone, ADH, which acts on the kidneys to prevent the loss of water via the urine. And then number 8, you have somatostatin. Somatostatin, it inhibits the release of growth hormone. So those are the 8 hormones released by the hypothalamus. If you do anatomy and physiology, you have to know this very well. And then, for your nursing career too, it's good to know these things very well. Okay, because you can have abnormalities of them. But the one way you can remember, as I said, is a releasing hormone attached to them. You know it's from the hypothalamus. Okay. So for the test, remember that hypothalamus produces hormones that stimulate the pituitary to produce other hormones. I think that should go a long way. Okay. So that's the hypothalamus. Then we have the pituitary gland, very good, famous gland that you should know about very well, just beneath the hypothalamus, somewhere here, beneath the hypothalamus. Okay. So some of the hormones released by the pituitary, we've mentioned them. We have the growth hormone, TSA, thyroid stimulating hormone. Let's go down so we can have a look at them. Okay. So here we have the pituitary gland. It has anterior portion, which you call the hormone producing glandular portion, and it has a posterior portion. So it has anterior. So you take a pituitary gland, This would be like the hypothalamus. It is then the pituitary gland will be hanging underneath it like this. And as we say, has two parts. So this is a pituitary. So this part is called anterior pituitary. And then posterior. Okay. So the anterior part is the hormone producing part. So this part produces the hormones. But the posterior part is known as a neural part. Neural part because you have like nerves that come in there from the hypothalamus. And it basically is like an extension of the hypothalamus. Okay. Now the posterior part does not produce hormones. It just stores hormones from the hypothalamus. Okay. So those two hormones are the oxytocin. We call the oxytocin and the ADH. Okay, so they are stored here, produced by the hypothalamus, but stored in the pituitary. Okay, so storage. Whilst anterior one produces hormones. Okay. And so I have the details written down here to help you remember. Okay. So what are the hormones produced by the pituitary? Okay. So 4 of the 6 pituitary hormones are called the tropic hormones. Tropic hormones. No. So they regulate the function of other endocrine glands. So the function of the pituitary is to produce hormones that regulate the function of other glands that we're going to talk about. Okay. So here are some of the hormones produced. The growth hormone. The name tells you what it does. So growth hormone, it stimulates the growth of all body tissues, especially the skeletal muscle bone and bone, right? Growth hormone mobilizes fat. It helps to mobilize fat. It stimulates protein synthesis, and also it inhibits glucose uptake and metabolism. Okay. Yeah. Now, overproduction can lead to what we call gigantism. So gigantism. Whilst underproduction will lead to dwarfism, like reduced growth, during the growth period. If when the person is growing, the growth period, rapid growth period, and if you have high levels, the person will become like a giant. You have low levels, then becomes like a dwarf. Okay. Now, if the person is already grown, grown up, and the person have sudden increase in like production of growth hormone, maybe from a tumor, okay, then the person cannot grow tall, but the person will be expanding laterally in the lateral direction, right? So you see the bones will become bigger, like laterally, sideways, and we call that acromegaly. Acromegaly. Okay. Then 2, it also produces thyroid stimulating hormone, TSH. So the thyroid stimulating hormone acts on the thyroid gland, right? So it promotes normal development activity of the thyroid gland. So thyroid stimulating hormone, TSH. Okay. You will learn about that again in your clinical courses. The level can be high or it can be low. When it's high, then the person develops what we call hyperthyroidism. If it's low, the person develops hypothyroidism. Okay. And one thing I would like to add here is the fact that you need iodine. Okay, so for this here, you need iodine is needed. Oh, no. Okay, let me leave that out. I'll talk about that and not on this level. I'll talk another when I talk about the thyroid gland. We'll come to the thyroid gland. I'll mention, that's what I'll have to talk about this. But TSH stimulates release of thyroid, the thyroxine, thyroid hormone. And then number 3, you have adrenocorticotropic hormone, ACTH. This stimulates the adrenal gland to release the corticosteroids. They produce the corticosteroids. We'll talk about that, and adrenal gland. Then you have the gonadotropin releasing gonadotropins, the gonadotropins. Example will be the follicle stimulating hormone, we call FSH, and they also have what we call the luteinizing hormone, LH. All right, so these regulate the functions of the gonads in both sexes. FSH stimulates the sex cell production, and LH stimulates the gonadal hormone production. Okay, we'll talk about the effect of this on the testis and then the ovary. Okay. Then prolactin, PRL, the prolactin, that stimulates milk production in human females. Okay, so those are the hormones that are produced by the anterior pituitary gland, the anterior pituitary gland. Okay, now the posterior part, as I said, just stores the hormones, and so that's what I have here. Oxytocin here, which stimulates so this stimulates powerful uterine contractions, contraction of the uterus during labor and delivery of babies. It also helps in expression of the milk, the ejection of the milk in nursing mothers. Then you have the antidiuretic hormone, which acts on the kidney. So it stimulates the kidney tubules to reabsorb and conserve water. This results in the production of small volumes of highly concentrated urine and decreased plasma osmolarity. Okay, so it helps you to retain water, so you don't lose too much water. Okay, so underproduction leads to a condition known as diabetes insipidus. So this is different from diabetes mellitus. That's two different things. This insipidus, that means they cannot concentrate their urine. They lose a lot of water, where the affected person passes a lot of diluted urine. If they don't have enough of this ADH, if it's not functioning well. Okay, so know some of the hormones produced by the pituitary. Okay, so the pituitary is described as the master of the orchestra. So when they say which organ is the master of the orchestra, you're referring to the pituitary gland. Okay, master of the orchestra. So it's like conducting, the orchestra. He's like a master. Do this, do that. Stop playing. Function. Stop functioning. And that's what it does to the hormones. The other glands. Okay. Okay. All right. So that is about the pituitary gland. Okay. So now let's see what it does to the other glands. Okay. So we look at the thyroid gland. So if you go back to the diagram, we know where thyroid gland is. Thyroid gland is right here. Thyroid gland around the anterior part of the neck. Okay. And then within that, you have a pair of glands on both sides of it. So those are known as the parathyroid gland. They're tiny, two on each side here, called the parathyroid gland. Okay. So that's why you have the two around the same area. Okay. So what's the function of the thyroid gland? Let's go down here. Now the thyroid gland, the function is to produce the thyroid hormone, right? And the thyroid hormone includes the thyroxine, which you call T4, and then T3, which is triiodothyronine. It increases the rate of cellular metabolism. Okay, so they control metabolic activities, right? So this one I'm going to talk about the role of the iodine. Okay, now you need iodine. Iodine is needed for the thyroxine production. Okay. You need iodine here. Okay, so triiodothyronine, that means 3 iodine in this one. So this has 3 iodine. The thyroxine has 4 iodine. That's called T4. So that one has 4 iodine in it. While this one T3 has, and the T4 is the active form. So this is the active form of the thyroid hormone, the one that functions. Okay, so T3 has to be changed to T4 for it to act on the body. Okay, so remember, iodine is needed for thyroxine production. So if you have a question where they tell you somebody lacks iodine in the diet, what's going to happen to the thyroid gland? You should answer that question. The thyroid gland is going to be deficient in its production of thyroxine. Okay. Now this is the reason why they put iodine in the salt, the iodinized salt. That's the reason. If somebody lacks that, then the person will have what we call hypothyroidism. Hypothyroidism. Those low levels, right? Low thyroxine. Okay. That's low production of a thyroid hormone. Okay. And what happens is that they develop what we call goiter. They have what we call goiter. Goiter is swelling of the thyroid gland. So thyroid gland ends up swelling. Okay, we don't see that a lot in this part of the world, but in other developing countries where there's deficiency of iodine, you see that the thyroid becomes big. It becomes very big. If you Google, you may see some of that. You see a lot of diagrams about that swollen thyroid gland. Okay, once they're treated with iodine, they get fine, and the swelling goes down, and then they start producing normal amounts. Okay. Now, what are some of the presentation? The clinical presentation here. I always like to stimulate your mind a little bit so you think ahead. Now, the person has low levels of thyroid hormone, the metabolic rate will go down, right? So the metabolic rate goes down, and then because of that, they don't produce a lot of heat. So you see that the room temperature will be okay. Everybody's okay with the normal room temperature. They are fine. But the person suddenly says that I'm feeling too cold, but they always feel very cold because they don't generate a lot of heat, so they always we turn the heat on because they're too cold. And other thing is that they also can concentrate very well. Okay, they have what we call very poor mentation. They can think very well because they're a little bit down, and moody. Okay. They don't eat a lot, yet they put on a lot of weight, all because of low metabolic activities. So that's what happens with the hypothyroid. Then you have the hyperthyroidism. For hyperthyroidism, they have the high production of the thyroid hormone or thyroxine. So that's high thyroxine levels. Okay. So how do they present? They will have a high metabolic activity. So they generate a lot of heat. So because of that, if the room temperature is okay for everybody, for them, the room will be too hot. So they'll tell you, can we have the air condition on? Please turn it on, Then they also are very anxious. And they also get irritated easily. In a small thing, they get so irritated. so they're irritable. So nothing's wrong, but if suddenly they can flap on you and so on, because they're irritated easily. Any small thing, they get irritated, all because of the effect of the high levels of the hormones. They eat a lot, but then they don't put on weight. They lose weight, because they have a high metabolic activity. So these are some of the presentation if you get into the clinical aspect of things. Okay. Yeah. So take note of these two: hypothyroidism, and goiter. Okay. Yeah. Then the other thing that the thyroid produces is this hormone known as calcitonin. Calcitonin. Okay. So calcitonin is produced by what we call the parafollicular cells of the thyroid gland. Okay. And the function is to decrease the blood level of calcium. So they decrease the blood level of calcium. And that's a function of the calcitonin. So if you see calcitonin question, it will reduce calcium level in the blood. Okay. Then the other hormone that we spoke about, other gland, the pair of glands that I pointed out, 4 of them in the thyroid gland, 2 on this side, the parathyroid. The function of parathyroid is to produce what we call the parathyroid hormone, PTH. And the function of the parathyroid hormone is to increase the calcium levels in the blood. Okay, calcium levels in the blood. Okay. Remember, calcium is stored in the bone. Calcium is stored in the bone. So if the level goes up in the blood, then calcitonin will cause it to be pushed or taken up into the bones. Okay. Whilst if there's low level in the blood, then your bone have to break down. The parathyroid hormone will break down the bone and cause it to release calcium into the blood. So that's how it goes. The storage in the bone, calcium storage, bone. Okay. All right. So those are the functions of the thyroid gland and the parathyroid glands. Okay. Then the other gland is the pancreas. If you go back to the diagram, you have the pancreas right across, right there, pancreas. So the pancreas produces insulin, glucagon, and somatostatin, right? Okay. But we'll concentrate on 2 main organs, 2 main glands here, the common one. So the pancreas is both an exocrine and endocrine. So you have to note this very important statement. That's the organ that has both exocrine function and endocrine function, right? We'll talk about the exocrine function when we talk about digestion, where it produces the digestive juices, So endocrine means has duct. I've said that already. The endocrine portion is known as the islet of Langerhans. Okay, and that's where you have the release of insulin and glucagon. They produce the insulin and glucagon. Okay, they are produced by the islets of Langerhans. Okay, now as I said, it also releases small amount of other hormones into the blood as well. Somatostatin, for example. Now let's look at the function of glucagon and insulin. We'll focus on these 2 main hormones, the common ones. Now, glucagon is released by what we call the alpha cells. And the function of glucagon is to increase the glucose level in the blood. So it increases glucose level in the blood. And then insulin is released by what we call the beta cells. The function of that is to reduce the sugar or glucose level in the blood. Okay. So how does it do this? It increases the rate of glucose uptake and metabolism by most of the body cells. So it causes your body to increase the use of glucose. Okay, that's what insulin does. Okay. If the level goes down in the blood, then glucagon will be released, and that will stimulate the release of glucose into the blood, like from the liver. The liver stores a lot of the glucose in the form of glycogen, So glycogen will be broken down to release glucose. So know the function of glucagon and insulin, at least for this particular gland, very common hormones. Okay. If insulin is not working well, then that's where you're going to have the diabetes mellitus. Okay. So this will be deficiency of insulin. Either it's deficient or not working well. So a deficiency of improper functioning. They have diabetes insipidus. Okay. Later on, you'll be talking about diabetes in your clinical practice. There are different types, of diabetes. Okay. All right. So those are the functions of the pancreas. Quick overview of it. Okay. Then we get to the gonads. The gonads are right there. So have the ovary and then the testis in the male. So those are the gonads. So what are the hormones produced by gonads? So the gonads, the ovaries of the female releases two main hormones, estrogen and progesterone. So estrogen and progesterone. Okay. Estrogen stimulates the maturation of female reproductive system and development of the secondary sexual characteristics, whilst progesterone works with estrogen in establishing the menstrual cycle, right? Then the testes of the male produce testosterone. So testosterone. Okay. Now this promotes maturation of the male reproductive organs, development of the secondary sexual characteristics, and production of sperm by the testes. Right. So that's a function of the testosterone. Okay. So those are the gonads. And then you have the pineal gland. The pineal gland somewhere around here. Pineal gland a little bit behind where the hypothalamus is, and it produces melatonin. It produces melatonin. Now, the function of melatonin is to regulate your sleep and wake patterns. Okay, so pineal gland produces the hormone melatonin, which influences the daily rhythms such as sleep and wake patterns. And I have to mention here that it's affected by the sun light or light. So it's always good to, if you work in the night, it's good to turn off the light a little bit for some period so that your body, adjusts itself. You need like that rhythm, day, night, pattern. Light off, light on. The night is very important to stimulate production of pineal gland. Otherwise, your sleep pattern becomes messed up. Okay. And then the other gland is thymus, the thymus gland. Thymus gland, it's somewhere behind the sternum. Thymus gland. All right. Now, the thymus gland usually vanishes by the time the person is born because it has already done its work. So we refer to that as a vestigial gland. It's a vestigial gland. It becomes very tiny, or almost gone. Okay. So it's an important gland of the immune system. during the developmental stages of life. It vanishes by the time of birth. By the time of birth. Now the function is this. It's the site where the T cells mature. They mature in the thymus gland. That's why I call it T cells. Now what happens is that the T cells, which are like lymphocytes, right? One group of lymphocytes, they go to school in the thymus gland. So we use the word the thymus gland. Schools the T cells. In other words, we are saying that it trains the T cells to recognize the self. It helps the T cells to recognize the self. What do you mean by this? Now, when the T cells are produced, initially they don't know your tissues. So they go to the thymus, and the thymus trains them to recognize your tissues so that you don't damage your own tissues. That's a function. So those who have autoimmune disease, it is said that, or one of the theories is that their T cells are not trained well by the thymus gland. That's why they're, like damaging their own tissues. That's one theory of the autoimmune disease. All right, so these are some of the hormones produced by the glands that we've spoken about. There is one more gland which I'm going to talk about. I think I didn't mention that. And I don't see that in my notes here. So let's go back to the diagram. I have it on the diagram. Okay. Now the adrenal gland, which is above the kidney, right? The adrenal gland, right there, adrenal gland. Now the adrenal gland produces the androgens, the hormones are called the androgens, and they also produce glucocorticoids, then adrenaline and noradrenaline. The adrenal gland has two parts. Adrenal gland. It has what we call the cortical part, the cortex, and then the inner part, cortex is on the outside, and then the inner portion is what we call the medulla. Has a cortex and medulla. Okay, so that's outer and the inner portion. The cortex, that's where you produce what we call the glucocorticoids. You produce the glucocorticoids and the mineralocorticoids. Glucocorticoids. And then the mineralocorticoids. Okay. Now the word tells you what it does. Gluco. This means they act on glucose metabolism. Right. The mineralocorticoids, they control like the secretion of minerals like sodium and potassium in the kidneys. They control how much is lost in the urine and so on. Okay. So the glucocorticoids, that's where you have what we call the steroid hormones, the cortisols. The stress hormones, the stress hormones, cortisol. So when something is stressful, they are released and they help you to deal with stress. They involve metabolism, generating energy by breaking down glucose and so on to give you to deal with the stress. Okay. Stress hormones. That's one way you can remember this. Okay. Yeah, mineralocorticoids, they have names. An example would be like what we call aldosterone. Aldosterone is an example. I don't think you need to know all this for the test, but just in case. So aldosterone acts on sodium, like causes sodium, control the sodium secretion in the kidneys. Okay. Then when you get the inner part, which is the medulla, that's where you release your adrenaline, and then the noradrenaline, what we call noradrenaline. They are released here. So they're also stress hormones. They help you deal with stress. Yeah. So those are the function of the adrenal gland. And then if you come to the stomach releases some hormones too. Some of the hormones released, I'll just mention them, like gastrin, histamine, somatostatin, neuropeptide, and so on. Okay. Some help you to manage pain. Some histamine, like dealing with allergic reactions and so on. Okay. But the ones I've spoken about are the common endocrine glands that you should know about. Okay. All right. So I think this should be enough for the endocrine gland. Any questions on that? Endocrine glands. All right, so I believe so far everybody's fine with that until we see some questions, right? Okay. Now let's talk about the digestive system, another system. Today I'm talking about a lot of systems. Digestive system. So this is a diagram of the digestive system that I drew some time ago. And there's one here too, this one. So the digestive system, another name for digestive system is the alimentary canal. You know, if you look on the test, they ask you that question. What's another name for the digestive system? And they have alimentary canal. So you see that word, you don't talk about digestive system. Okay. And it starts with the pharynx. The pharynx. Okay. So you have the pharynx, the mouth, you have the mouth that opens into the pharynx first. Okay. And then you have the esophagus into the stomach, right there, stomach. And then from the stomach, you go to the duodenum. Somewhere, the duodenum is going to be somewhere here. A small intestine, first part of the small intestine, and they have the other part of the small intestine, which is the ileum and the jejunum, somewhere around there. And they have the large intestine, the large intestine. You have different parts of the large intestine. The ascending, transverse, and a descending portion of the large intestine right there. Okay. And then you have the sigmoid colon. Another name for the large intestine is the colon. The colon. Okay. So here the sigmoid colon, and they have the rectum and the anus here. Okay. Now know what happens in the different parts of the intestine. So and the digestive system. Okay. So let's begin with the mouth. Now, if you take the mouth, now in the mouth, that's why you have breakdown of the food. So that's mechanical digestion. We call that mechanical digestion, right? So the teeth breaks down the food into small pieces, and then chemical digestion starts also in the mouth. Because the saliva contains an enzyme we call the amylase. So you have your amylase in the mouth. So the amylase, the function is to break down starch into sugars. It breaks down starch into sugars. So know this very well. So this is where the question comes in. If somebody chews crackers slowly, what happens to the taste? How does it taste if you chew crackers slowly? They have the where it says it's salty, sweet, bland, bitter, and so on. So starch has a bland taste. But when you chew slowly, the amylase will break down the starch and change it to simple sugars like glucose. And this is what will taste sweet. So it's going to taste sweet when you chew slowly because of chemical breakdown. Okay. Then the tongue will roll the food into what we call a bolus. The bolus. So you have to know what a bolus is. Bolus is the food that is rolled into like a ball of the food. Okay. Give me a second, sorry. So you have the bolus that goes down the esophagus. The tongue pushes it back into the pharynx, and then goes down into the esophagus. Okay. So there is this flap over here called the epiglottis. The epiglottis is like a muzzle flap of muzzle that closes over the larynx. It prevents food from going into the what we call the trachea. Okay. So in other words, direct food into the esophagus. Okay. So the bolus goes through the esophagus by means of a process known as peristalsis. So remember, when you get to the esophagus, the word peristalsis. Okay. So remember peristalsis here. So peristalsis is the rhythmic contraction and relaxation of the muscles, of the esophagus. Okay, that propels the food down. It pushes the food down into the stomach. Okay, so it's like a snake moving. It's like a snake-like movement that pushes the food down into the stomach. Peristalsis. Okay. So once the food goes in the stomach, we need to know what happens in the stomach. So in the stomach, you also have what we call mechanical digestion. The smooth muscle contracting, relaxing. It breaks down the food further, right? And also mixes it up with the gastric juice. So here you have gastric juice in the stomach. Okay. It makes the food more watery. Then you have chemical breakdown. The chemical breakdown in the stomach, you have the enzyme known as pepsin. So you have the pepsin. Know about the pepsin. Pepsin is in the inactive form known as pepsinogen. This inactive enzyme, pepsinogen. Now this is changed to pepsin, which is the active form of the enzyme, pepsin. And you need the acid, HCl, hydrochloric acid. So the acid in the stomach helps to stimulate or activate the pepsinogen to become pepsin. And what's the function of pepsin? Pepsin breaks down, this is what breaks down proteins. Protein breakdown. Or you can say digest proteins. It helps to digest protein. Okay. So protein digestion, in the stomach because of the presence of the pepsin. Okay. Now another function of the hydrogen chloride, HCl acid, is to kill bacteria. Also kills bacteria in our food. So we have HCl, the acid. Also breaks down bacteria, kills bacteria. Then the other thing you have in the stomach is the mucus. Produces mucus. Okay. And the mucus protects the lining of the stomach. It protects the lining. Okay. It's very acidic. So the lining is produced. If the mucus is not rich or something like very good, then the acid can digest or break down the lining of the stomach. And then people develop ulcer. That's how you get the peptic ulcer, like sore in the stomach, which is very, very painful. So you talk about ulcer in your clinical practice as well.
I'm sorry, can you explain what the mucus does
The
one more
mucus,
time?
okay, the mucus, it's like slimy, and then it protects the stomach lining. So it's like covers the surface of the stomach so that the acid doesn't, damage the epithelial tissue. So what I was saying is that if the mucus is not produced well, then the acid can have access to the underlying tissue and then to damage it, right? So once it damages it, it causes like a sore, which we call the ulcer. Peptic ulcer, that's the name, peptic ulcer. Okay. All right. So that is what you have in the stomach. Okay. I mean, you also have some small amount of like lipase that can break down lipids. Yeah. But the bulk is like the pepsinogen. Okay. So once the gastric juice, the juice produced by the stomach lining, called gastric juice, mixes up with the food, it forms what we call the chyme. So chyme, this word here, chyme, CHYME. Okay. It's a mixture of the gastric juice and then the food, the raw material, which is the food being digested. Okay. It becomes more watery, and then it releases into the first part of the small intestine, which is called the duodenum. So the duodenum is going to be somewhere around here like that. If I put the red and put the red mark, okay, the duodenum will be somewhere around there. So the small intestine, like a small intestine, has different parts. The first part is the duodenum. The second part is known as the jejunum. And the third part is known as the ileum. So those are three parts of the small intestine. Okay. So the duodenum, what happens in the duodenum? The duodenum, that is the part that you have the release of the bile. Bile is released in here. Okay. Because if you see the duodenum, it's going to look something like this. You have, it is the stomach. Let's say if you draw the stomach like this. This will be the duodenum. Okay. The pancreas will be somewhere here. This will be the pancreas will be somewhere here. And you have the gallbladder will be somewhere here. Gallbladder will be here. And then it opens into the duodenum around here. The pancreas will be somewhere there like this, pancreas. Okay. So the gallbladder is here, gallbladder. Okay. So it releases the bile into the duodenum right there. This is the liver. Okay. Then the pancreas also releases what we call the pancreatic juice. So bile is released here. pancreatic juice is also released there. Pancreatic juice. Also released here. So it's a very important area. And the pancreatic juice will contain enzymes, right? Usually contains all the different enzymes that can help to digest carbohydrates, proteins, and fats. So we say that you have amylase, you have lipases that break down lipids, and then you have proteases. These are all hormones that are released by the pancreas that help to continue digestion of the food. Okay. So now it's important to talk about what happens and what the bowel does. Okay, the bowel. What's the function of the bowel? The function of the bowel is to emulsify fat. It emulsifies fat. That means it helps to break down fat into small droplets. If you take fat cannot dissolve in oil, in water, right? It doesn't dissolve in water. But then if you use soap, something like soap can help the fat, mix up with the water, okay, or break it apart into the water. The same thing is what the bowel does. The bowel helps to break down the fat into small droplets in water. It helps so it can mix with water. So we use the word it emulsifies. I'll write it down for you soon. Okay. So the bowel is very important in digestion of fat. Okay. Then the jejunum, digestion continues the jejunum, the second part of the small intestine. And then when it comes to the large part of the small, the longest part of the small intestine, which is the jejunum. The jejunum, this is where you have a very important structure known as the villi. So you have the villi. This also comes up on the test a lot. Villi. Okay. Singular is villus. Villus singular. Villus. That's singular. Okay. So the function of the villus is to increase. So that's very important. I'll put asterisks against that. You're going to come across that a lot. So the villi increase the surface area for absorption of food. Absorption. Of the digested food. Okay, so that's the function of the villi. Okay, so put asterisks against this if you're writing down notes. Very important. Okay. In other words, without the villi, then you have a small area for food to absorb. Okay, you see, one way you can look at the villi, think about the towel. If you take out the towel, the threads standing out, those are like villi. So they're all increasing the area for absorption. Okay, so they're packed in one small area, is packed, but then each surface is used to absorb something. So that's a function of the villi. Okay. All right. And then you get to the liver. The liver, that's where I want to mention the fact that the gallbladder stores bile. So let's look at the liver. The liver has many functions, so many functions. Okay, so one of the function is to store starch. No, I said starch. Store glycogen. We don't have starch in the human beings, so it stores glycogen. So it stores excess glucose as glycogen. So that's how we store our extra glycogen. Very important to know this. And then it's also involved in storage of lipid metabolism. Lipid metabolism, and also protein synthesis. Protein metabolism. Another function of the liver, a very important function, is detoxification. Detoxification gets rid of toxins, waste product, detoxification. That's another function. Okay. It also stores a large amount of blood too. You know, and it also breakdown of the red blood cells. Red blood cell breakdown. Okay. So good to know this. And then important one, production of bile. Bile production. Bile production. So we're going to look at the function of the bile. Now, the gallbladder. So remember that the gallbladder just stores the bile. It doesn't produce bile, but it stores bile. The bile is produced by the liver, stored by the gallbladder. Okay. So it's almost like golden yellowish color. The bile looks like golden yellowish type of color, or something a little greenish. And what's the function? The function of bile, as I said, bile helps. It helps to emulsify fats. Helps to emulsify fats. In other words, helps to break down fats into small droplets in water. But remember that the pancreatic juice has a lot of water there, you know, so it helps the fat to mix up. Now, another important thing about the gallbladder, we know that there are some fat-soluble vitamins, right? The fat-soluble vitamins. Now, these are vitamin A, D, E, and K. So we say ADEK. That's the way we can remember ADEK. The ADEK, A, D, E, K. These fat-soluble vitamins need bile to be able to get absorbed very well because of fat, and they need the fats. So fat needs bile, you know. So if you take somebody's gallbladder away, you take it out during surgery, for example, in what we call, we use the name for the surgery is called cholecystectomy. In cholecystectomy, if you take out the gallbladder, then the person is going to have problems absorbing these fat-soluble vitamins, you know. So you need to give them some supplements. Okay. So it's very important to know these fat-soluble vitamins. Okay. So it helps in absorption. Bile helps in the absorption here. Okay. And then the last part of the intestine will be the colon. The colon. So the colon, that is where you have the large intestine. That's where you have storage of the feces. So whatever is not absorbed is released into the colon, and that is the feces. Okay, so that is the summary of what is happening in this diagram. Appendix usually has no particular function. It's very unnecessary. It doesn't it really causes problem. But it's thought to be part of the lymphoid system, like fight infection. The lymphoid system, they produce, they help to fight infection. Okay, but it's not a necessary organ in the body. Rather, causes when it's blocked, it can get inflamed and cause problems. You know. Okay. And that's what we call appendicitis. Okay. So this is a quick overview. You know, I have everything written down here for you and typed out here. If you read that, whatever I spoke about, they're all here. Carbohydrate digestion begins in the mouth, enzyme amylase present, blah, 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 all those things I spoke about. Okay. So they are all here. So take your time to go through the handouts, and then you see that whatever I spoke about here is all typed in here for you. Okay. All the key points. All right. Any questions on the digestive system? These are big, big topics, you know. All right. We'll look at another system. Now, the other system, it is a genitourinary system, the genitourinary system. So the genitourinary system consists of the kidneys. So you have the kidneys, left and right. Then you have the ureter that collects the urine from the kidneys into the bladder. And then bladder, from the bladder, you have the urethra. So those are the components: ureter, urethra, bladder. Get the bladder here, and the kidneys. Now, what is the basic unit of the genitourinary system? The basic unit is known as a nephron. So we're going to look at a nephron. So they like to ask what's the basic unit. So basic functional unit, called the functional unit of the kidney. Is the nephron. Nephron. Okay. And this is the structure of the nephron. It looks like this. Okay. So basically, if you look a simple diagram, or draw a simple diagram, it looks like this. And they have a first part that goes like that. And they have a hairpin loop. Makes a hairpin loop, and then another curvy part like this, and then collecting that. This is how it looks like. Just redrawing the diagram. And they have blood vessels coming in. Blood vessels come in, break down to a network of blood vessels, and then they leave. Okay. So this blood vessel here is what called the efferent efferent vessel. So we talk about here. These are the blood vessels coming in here, breaks down to a network of blood vessels, and then leaves right there. Okay, so this vessel is not the afferent vessel or arteriole. That's the efferent. efferent vessel or arteriole. Then the one that goes out is known as the efferent vessel or the arteriole, efferent arteriole that goes out here. Then the breakdown of the blood vessel into capillaries, the capillaries that you see here is what we call the glomerulus. Glomerulus. So this here is a glomerulus. Glomerulus. Okay. So in the glomerulus, that's why you have filtration of the blood. So the blood goes in and is filtered. So this blood gets from here, goes in, and then it's filtered here before it leaves out. So once it filters out, you have what we call a filtrate. The filtrate collects in here. Okay. And the cap-like structure that holds the glomerulus, this here is known as the Bowman's capsule. So it's like a cap holding the capillary, the glomerulus, Bowman's capsule. So this is your Bowman's capsule, all this here. And then the first part of the tubule is known, this here goes up and down. What you see going up and down here is known as a proximal convoluted tubule. So this is the word proximal convoluted tubule. Okay, so this here is a proximal convoluted. Convoluted means like curvy, tubule. So that's where the filtrate collects. Okay. And then once the filtrate collects, it flows through the loop here is known as a loop of Henle. Loop of Henle. Somebody's name, loop of Henle. Okay. And then over here, we have what we call the distal convoluted tubule. Okay. Distal convoluted tubule. And at the end here, what you have is called, this part here is called a collecting duct. This is a collecting duct, and that's what is here, collecting duct. Okay, so those are the parts of the nephron. Okay, the part of the nephron that, as you see in the diagram. Okay, so what happens in the nephron? Very important to know what happens in the nephron. This is where the urine is produced, the dry urine is produced. And the process of urine production, there are three components of it, three components of urine production. So let's write that down. Urine production, because there's a question that asks you about that somewhere, I think the book. So the first step in urine production is what we call filtration. You filter the blood. And the second step is known as reabsorption. Reabsorption. And the third part is known as secretion. So those are three steps involved in the production of urine: filtration, reabsorption, and secretion. Okay, so let's go back to the diagram for you to follow what goes on here. So I guess started with the first part where I said the blood is filtered in the Bowman's capsule, right? Passes through glomerulus, filtered here. So that's the first part. And then once you're filtered, so that's here. Step one is this step one. Production of the filtrates. Okay, it's like a filter paper, if you know filtrate. So not everything goes into a filtrate. You can have like the red blood cells. They are too large. White blood cells, all the cells in them, they are too large to pass through the glomerulus into the proximal convoluted tubule. Okay, so they don't go out. You only have part of the what do you call it, plasma, the liquid part of plasma. That is what goes filtered out. We dissolve substances like glucose, and other ions dissolved in there. They go into the proximal tubule. So if you see red blood cells in the urine or white blood cells in the urine, then there's a problem with the glomerulus when you're doing clinical practice. You don't expect them to see them in the urine unless there's a problem, a leakage in the glomerulus. Then you have those big cells in there. Okay, then it means there's a disease going on. Okay, so when you get to the proximal convoluted tubule, that's where you have this number 2. This is where you have the reabsorption of urine. Sorry, I said reabsorption. Sorry, I'm using the wrong thing. Reabsorption of water. You have the reabsorption of water and ions and all organic nutrients. They all occur in the proximal convoluted tubule. So as we said, they are occurring here. Reabsorption occurs there, okay, of the good part of the filtrate. Okay. Then as it passes through the loop of Henle, you have different parts of it. You have descending loop. These are descending. So these are descending parts. Then you have ascending parts. That is descending right there and ascending right here. Okay. So here, this word you have further reabsorption of water. Both sodium and chlorine are also absorbed in the ascending limb. So here you have sodium goes out along this part here. Sodium goes out here, and chlorine, they are released in the ascending part, okay, into the surrounding tissue. Then as the filter flows down, the water is reabsorbed into the tissue. You know, we have something called countercurrent mechanism here that allows the water to be reabsorbed back into the tissues. Okay. So by the time it gets here, in the distal convoluted tubule, you would have a lot of concentration of the urine already occurring, right? So in the distal convoluted tubule, that's where we have the third step. This here is the third step, and that is known as the secretion. Secretion. So you release ions into the urine that is being produced. So here you have secretion of ions and then acids and drugs or toxins. They are secreted, means released from the tissues into the urine. Okay. And they have a variable reabsorption of water, sodium and calcium also occur under hormonal control. So this is where the hormones, the aldosterone, the mineralocorticoids, that as I mentioned, like aldosterone, this is where they play a role to control how much sodium goes in and out of the cell into the urine. Okay, all occur here, so have secretion under the hormonal control. Okay, and then you also have the ADH. ADH controls how much, as we said, under the endocrine system, it controls urine production, how much water you absorb. It also acts in the distal convoluted tubule to cause water to be absorbed. So those are the processes that occur. So concentration of the urine will occur right there. Further concentration occurs right here. And once the urine is produced, it collects in the collecting duct. This collecting duct that goes to the bladder. You don't go to a bladder, you know. So many collecting ducts will open into the ureters, and then it will go to the bladder. So that is the overview of how urine is produced. Filtration, reabsorption, and secretion. Those are the processes, 3 processes involved in urine production. Okay. So I believe this should be enough for urine production to answer questions on that. Okay. All right. Any questions on that one? And we'll talk about the last system. Any questions on urine production? Okay. You can ask questions as we go along. You can stop me. Okay. All right. So I think we spoke about the blood a little bit, if my memory serves me right. Okay. So this is a diagram of what makes up the blood. plasma form cells. I think we spoke about that some time ago, so we just brush over it. We have red blood cells, white blood cell platelets. I spoke about the different type of white blood cells and their function. So these are just notes for you to use. Okay. So I'll move on to the cardiovascular system. So this is a diagram of the cardiovascular system. Usually when I'm teaching this, I like to use like a box to illustrate the heart. And this can help you in clinical practice. There are some people I taught many years ago, they still have this diagram. They say, I still have your diagram. I say, yeah, that's a good diagram to use. It helps you predict a lot of things in clinical practice. So I'll use a box to represent the chambers of the heart. As we know, we have left side, right side. Okay. So if somebody is facing you, it's like opposite, it's like a reflection. The left becomes a right, right becomes a left. So this is a left. Okay, somebody, Melissa, have your hand raised.
I'm
Hello,
sorry. That was accident. I'm sorry, Professor.
Okay, no problem. Okay. Okay, so the left side of there's a left atrium. And then you have the left ventricle. Okay. Then we have the right atrium, and then the right ventricle. Okay. Then I'll put the lung somewhere here. I'll draw the lung. The lungs here, left and right parts. So let's go lung. All right. So we have blood that goes from the head region, comes all the way down, and that opens into the right atrium. And they have blood that comes from different parts of the body that also open into the right atrium. So I'm going to use the yellow. Let me change this, maybe a blue highlighter. Okay. So blood comes in like this, and the blood from the lower part of the body, and they all go through the right atrium. Okay, through the right atrium. Okay. Now the vessel here is known as the from the head region, that is superior vena cava. And then from the lower part of the body, you have the inferior vena cava. Okay. So the blood that is coming in is deoxygenated blood, blood without reduced oxygen or without oxygen. So this is deoxygenated blood on that side. Okay. Because the oxygen has been used by the tissues. So once it gets to the right atrium, it flows through. There's the a valve here. There's a valve here, valve that controls the flow of blood, to prevent the blood from going backwards. And that valve is the tricuspid valve. So this valve here is the tricuspid valve. Okay. So the blood goes in to the right ventricle, and then from the right ventricle, the blood will flow out to the lung. The blood going go to the lung. Okay. So all this again is deoxygenated blood that is going into the lung. Now this vessel that takes blood to the lung is known as a pulmonary artery. Pulmonary artery. So oxygen is added to it in the lung, to the blood in the lung, and then the blood flows back to the left atrium through a vessel, another vessel, which is the pulmonary. So this is a pulmonary vein. Okay. So I'm going to use a red marker to highlighter to show you the direction of flow. Then the blood flows in through the valve here, which is known as the bicuspid valve. Okay. Bi means two, like leaflets. Tri means three, leaflet or cusp. So the blood goes in, use a red highlighter. So the oxygenated blood comes in like that into this and flows here, and then comes out through another artery known as the aorta. So this is a schematic diagram that will help you to understand what's going on. So the blood flows to different parts of the body, or different parts of the body, make it so many vessels all over the place like that. It goes to the tissues. And then this from the tissues, veins that go to the vena cava. Something like this. So these are different parts of the body. Okay. All right, so that's a schematic representation. Okay. Now the circulation over here from the lung to the right, to the left atrium, and then right atrium to the lung. This part here is not like a pulmonary. Pulmonary has to do with from lung to the heart, from heart to the lung. We call that pulmonary circulation. Pulmonary circulation. And then the part that goes from the part that's the left ventricle all the way to the different parts of the body, and then from different parts of the body to the right side, that circulation is known as systemic. Systemic circulation. Okay, so heart to the body, body to heart. Circulation. Okay, so that's a schematic diagram of what you see on the right side, on the left side here. Okay. The boxes helps you understand it easier. So what can happen here? If you're doing clinical practice, you'll be talking about heart failure and so on, heart failure. So heart failure, that means the heart has failed to pump. It cannot pump the blood out very well. There's something wrong with the heart. So let's say the left side of the heart fails, we can use this diagram to predict what's going to happen. Okay, so if this part is not working, let's say the left heart is not working well, they have left heart failure. So this means that this side is not working, problem with this part of the heart. What's going to happen? It means that the blood is coming in, but it cannot flow out. It's not pumping out. So what happens? You have the blood going backwards. We call it pooling of blood backwards, like a blockage. There's a block here, so the blood cannot go. So everything goes back. The lung becomes filled with fluid, and that's what we call pulmonary edema. If this is not treated, to go in backlog, you're going to go fill up, and then eventually the right side of the heart will also fail. And then this will fail backwards, So if the right heart failed, what's going to happen? It means blood that's pumped in cannot go out. So again, there's pulling of blood backwards. Nothing is like a backlog. Think about traffic, the backlog, nothing's leaving, so it's backlog. So what's going to happen? Right heart failure. Then there'll be swelling of the legs. There'll be fluid in the abdomen. That's what we call pedal edema on the legs, and the abdominal edema, and so on. Ascites, called ascites. Fluid will be all over the person's tummy and the leg. If you see that, then you know I'm dealing with left heart problem. Okay. If there's fluid in the lung, they are dealing with a problem with the left side of the heart, and so on. If you see fluid in the lung and the fluid in the legs, swelling of the leg and the tummy, and so on, then you are dealing with combined heart failure. I mean, both hearts have failed, and so on. So this diagram can use you to, analyze those type of clinical situations. So the person comes and then they feel like they can't breathe very well. They have to wake up in the night and, they sit close to the window as if they are dying. They feel like they're dying, shortage of air, and shortness of breath, and so on. If you hear those things, they should think about the problem of the heart, for example. All right, so this is a quick overview of what you see in the cardiovascular system. Now, one thing I want to highlight or mention is the fact that you see the pulmonary artery. It's good to know the exceptions, and the pulmonary vein. Now, the function of arteries, they transport blood away from tissue. That's the function of arteries, right? So we can write a little note. All arteries, if you're doing anatomy physiology, you like to ask those questions. All arteries carry oxygenated blood. Except, so this is an exception, except the pulmonary vein. Or I said the pulmonary vein. Sorry, I'm talking about artery, sorry. I said the pulmonary artery. Pulmonary artery. So it's an exception. Pulmonary artery is an exception, right? Carrying deoxygenated blood. The same with the vein. All veins carry deoxygenated blood. Except the pulmonary vein. Okay, so the pulmonary vein is carrying oxygenated blood. If you look there, the oxygen has been added to it. So this is oxygenated blood on the left side, oxygenated blood, because it's coming from the lung. Okay, but the reason why it's a vein, because veins carry blood away. So it's carried away from the tissue. So it's a vein. This is carrying blood to tissue. Arteries carry blood to tissues. So it's supposed to be an artery, but then it's doing carrying a deoxygenated blood while it's carrying oxygenated blood. Okay, exceptions. So they are good for multiple choice questions. Okay. Any question on the blood? I'll stop on this particular one. Okay. So a quick overview. Okay. Okay. All right, we're making very good progress. All right. So that brings me to the end of the systems, the different systems that I want to talk about. Okay. All right. Now, last time we spoke about data analysis, how to analyze data. So this is just a summary of what we spoke about. And when we go through the book, we'll still practice with some of the data analysis. As I said, the answers are always in the question when you see those type of problems. Okay. So this is just a summary. You should to analyze, you should to interpret, and that graphs and pictograms, you should to make conclusions and observations, okay. Hypothesis, state hypothesis, and so on. Okay. And then we also spoke about the experimental groups, the different, like having control group. I remember I mentioned that it's an experimental group, the group that receives the treatment or the independent variable, and then the control group is a group that serves as a standard of comparison or a constant. The independent variable is either not present or set at a standard value. Okay, so that is what it's about. For example, you have plants that receive fertilizer. You want to compare whether fertilizer is good for the growth of plants. You have the plant that receive the fertilizer, give them fertilizer, that will be the experimental group. And then the control group will be the one that you set up without fertilizer. Then let them grow. After some time, you observe and see what's happening. If those that receive the fertilizer grow better than those that did not receive it, then you can make a conclusion that the fertilizer is at least, doing something. Okay. So that is the experiments like this. Okay. So you can control group, you can, like this one, you can use just plain water, the ones that are giving fertilizer. You can also do that. 2 groups, fertilizer, the other one, just water, and see how they grow after maybe 3 weeks or 4 weeks. Yeah. So you should identify those type of problems in questions. Okay. Okay. Right. So now let's look at another part of the lesson. I sent you 2 different handouts, right? So the second handout is what I've entitled lesson 4. So if you have that, then you can look at that as well. Okay. So in that one, we have the part of the flower, Okay. So we want to look at the flower parts. And I always like to use the hibiscus flower as a typical example. Okay. Now, know that the flower always have, they have male parts and female parts. They have male parts and female parts. Now, this may be on the same flower or they may be on different flowers. When you have both on the same flower, then we call that a monoecious plant. If they are on different flowers, called dioecious plants. Okay, those are some names that may come up in biology. Okay, so below you have 2 typical flowers here. Some flowers can look like this, the one they have on the left side, and some, like the hibiscus, looks like that. Okay, so but the basic thing, you have male and female parts. Now the female parts, let's have the male part here. Point number 1. The male part of the flower is what we call the stamen. The stamen is the male part of the flower, and it consists of the anthers and the filaments. The anthers and filaments, that's what you have right there. If I take hibiscus, the anthers and filaments will be here. Okay, this yellow, the filament is the stalk that holds onto that. So let me redraw that quickly here for you to see. So it is a flower, you cut across the flower, longitudinal section. So I'm going to have a structure that resembles this. Okay. And then inside you have And then here you have these structures here like that. Okay, so this is like the hibiscus flower that you see the right one here. So this is what we call the anthers. The anthers here. Anthers, anther. The anthers. So the anther is what contains the pollen. So this contains the pollen grain, the pollen. Okay. And then this structure that holds the anther is called the filament. Okay, that's why you see this here, anther and filaments. Okay. When you come here, if the flower is like this, then is the anther and there's a filament holding. Okay. So they form the male part of the flower, the male part of the flower, which is called the stamen. Stamen right there. Okay. So the anthers contain the pollen grains. Now the female part of the flower is what we call the pistil. The pistil. So the pistil is the female is the pistil. So if you have just a male part on the flower, then the flower is called a stamen. A staminate. Staminate. If your only female part of the flower, we call that a pistillate. Pistillate just the female only. Okay. And the pistil consists of what we call the stigma, the style, and the ovary and the ovule. Stigma, style, ovary, and ovule. So if you come to a diagram here, you can see nicely labeled on the diagram right there. That is the node. And then if you come here, the same thing. Okay, so on my diagram, then you have this is a stigma. The style is this long structure here, style. This is the ovary. And then the eggs within the ovary is called the ovule. The ovule. Okay. So those form the female part known as the pistil. Okay. This is the flower, the corona or the petal. The petal, the colorful parts together form the corona. And then the flower stalk. And these are the the calyx, called the calyx. Calyx. Like that's a receptacle that holds the ovary. And the sepals will be like those leaf-like structures on the calyx called the sepals. Okay, they fuse to form the calyx. All right. So the stigma is the sticky part where the pollen that attracts the pollen, right? So this stigma here, this is sticky, the sticky part of the plant, very sticky. So in what we call pollination, in pollination, you have the pollen transferred from the anthers. So the anther, from the anthers here, you have pollen transferred on here. So let me use a different color. So you have the pollen will fall on this sticky part. So that's the pollen that falls here. So when the pollen falls here, what happens? It's going to grow. It's going to grow through the style and then to go and fertilize the ovule. Okay, so that is what we call pollination. Pollination. So pollination basically is a transfer of the pollen from the anthers to the stigma. That's what we call pollination. transfer of pollen from anthers to the stigma. It may be a self-pollination or it can have what we call cross-pollination, right? Self-pollination involves the same flower, so it transferred from the anthers to the stigma, same flower. Or you can have cross-pollination, where it goes to a different flower. Okay, cross-pollination is very good because it helps to produce different, like a hybrid. You know, it's more stable, it's better. Like the cross-pollination is always better. You can have new characteristics that help the plant to survive harsh conditions. You know, now the pollen grows through the style to the ovary to fertilize it. Now, fertilization will always lead to seed production. You know, you have seed production and embryo formation. So the fertilized seed is the seed is the fertilized ovule, right? You fertilize the ovule, you get a seed. So remember that pollination, very important. There's a question that you have to think about it. Pollination leads to fertilization, because fertilization is the ovule plus the pollen. The pollen is like the sperm fertilizing the egg, which is the ovule. So you have fertilization. Now the fertilization will now lead to what? Zygote formation. Zygote to divide to find the embryo, and then that grows to be the seedling. Okay, seedling. So basically the zygote is like the seed. Okay, you put the seed. So note here that, so here we see that if there's no fertilization, then there will not be, if there's no pollination, you cannot have fertilization. If there's no fertilization, there will be no seed production. You have to note the trend, you know. So there's a question that asks you something similar to this. If all the bees in an area are destroyed, what is likely to happen? And they give you options they have to think about, you know. There'll be reduced seed production and so on, blah, blah, blah. And you know there'll be reduced seed production because no pollination, no fertilization. No fertilization, no seed production. That's how your mind should be going. Okay. Yeah. And they're agents of pollination, like bees, butterflies, human beings, air, wind. You know, these are all agents of pollination, things that can bring about pollination, you know. Okay. So I have a summary of what I'm speaking about here. Okay. But before I go on, I forgot to mention number 5 here. Said the pollen grows through the style to the, I said that. Then 6, fertilization leads to seed production and embryo formation. So the seed is a fertilized ovule. Then 7, the petals are the colorful parts, colorful, and they attract the agents of pollination, such as bees and butterflies, etc. The ovary wall becomes the fleshy part of the fruit, right? So the wall, the ovary, becomes the fruit that we eat, the fleshy part, like the wall of the ovary. Okay. And then I'll describe what pollination is all about. So that's the summary of pollination here. You know, so pollination leads to fertilization, and that's the male gametes in the pollen grain fuses with the female gametes in the ovule. The fertile ovule becomes a seed containing the embryo. The embryo germinates into the seedling. Okay, so that's the summary of what I put here. Okay. And then once you've gone through this, you should know about what do you call it, this seed. How the seed looks like. And the seed. Okay. So this diagram of the seed, very important to know. Take a look at it. So the seed has the outer covering, which is known as the seed coat. The seed coat. Okay, so that's the seed coat here around, you know. And the bulk of the seed, that you see shaded here, the bulk over here, is on the endosperm. Endosperm. So the endosperm is basically like a stored food for germination. It's mainly starch. okay, to produce energy for germination. The bulk of the seed is the endosperm. Then you have the small seedling within the seed, and that has different parts. The part that becomes the leaf is called the cotyledon. So the cotyledon here, I call the seed leaf. So it's a part that grows to become the leaf. Then you have the epicotyl. Epi means above, right? Above the cotyledon. So the part that's called epicotyl, the epicotyl is the part that becomes the shoot, the stem, and so on. And then radicle, the radicle is the part that becomes the roots. It grows to the roots. Okay. Another name for the radicle is called the hypocotyl. Sometimes they label hypocotyl. Hypocotyl. That refers to the radicle, you know. And then another name for the epicotyl, they use the word plumule. Plumule. If you see plumule, the same as epicotyl. Okay. Then another name for the seed coat, if you see the word tester, it refers to the seed coat, tester. Okay. So this is a quick diagram of the Bailey seed, for example. It's a typical example of how the seed looks like. Okay. So there's I think there's a question somewhere in the book where they give you the diagram and it's labeled 1, 2, 3. You have to identify the different parts and what they do. Okay. Great. So that should be enough of pollination and how the seed looks like. Yeah. Okay. We're going to switch a little bit to physics. I'm mind to some physics now. Okay. All right. So we look at how heat is transferred from one substance to one material to the other. Mode of heat transfer. Mode of heat transfer. Very easy to remember this. Okay. So we know that heat is a form of energy, and it can transfer from between bodies, which are under thermal interactions. Now, heat transfer occurs when there's a temperature difference between two bodies or a body with its surroundings. Right. So there should be some difference in temperatures between the two objects that are in contact with each other, you know, or they may not even be in contact with each other, but there should be temperature difference. Okay. And there are 3 modes of heat transfer. 1 is known as conduction, 2 is convection, and 3 is radiation. Radiation. 3 modes of heat transfer. So let's look at the details. Heat transfer. The first one, conduction. Now, conduction is heat transfer by means of molecular agitation within a material without any motion of the material as a whole. If one end of a metal rod is at a higher temperature, then energy will be transferred down the rod toward the colder end because of the higher speed, because the higher speed particles will collide with the slower ones with a net transfer of energy to the slower ones. Okay. So in conduction, what we are saying is that you should have 2 bodies, thermal bodies. So let's say I have 2 iron rods, hot and a cold one. So there's a temperature difference between the 2. Okay. Once they're in contact, the hot one, the electrons vibrate because they're high energy, so they keep vibrating. So they bombard against each other, and as they bombard, they transfer heat from one from one electron to the other. So that's what will happen in this iron rod, and then it's going to pass the energy on to the electrons in the colder one, okay, due to vibrations of the electrons. Okay. So see the bulk of the whole thing doesn't move. The It is an iron rod. Okay. The whole material doesn't move. It's just the electrons which are vibrating inside, the transferring energy from one form to the other. So that is conduction. It means there should be a contact between the two objects for that to occur. Okay. Now, in convection is the transfer of heat by currents. The key word is by currents within the fluid. So convection occurs within fluids. When I say fluid, we are talking about air. It's a fluid. It can move, particles can move fluid freely. Water is fluid. So it may arise from temperature differences, either within the fluid or between the fluid and its boundary, which will affect the density. Okay, so we're talking about convection occurring in a fluid. So this is what happens. Let's say this water, I have heat over here. So let's put on fire. So the particles that are lower down in the water, they get warmed up, they heat up. As they heat up, what happens? They tend to move faster. They gain energy and move faster. So what happens? They rise up. They rise, the fluid here will rise up, and then the cold water on the surface will descend because the density has changed. The density, if you heat it up, the density becomes lighter, so it moves up. And the heavy or the denser part sinks, goes down. Okay, so this will keep going on until the whole water is warmed up. So this is a current being generated in the fluid or the water. Okay, so that's what transferring the energy by means of currents, and that's called convection. Okay. The same thing happens in air. That's how we heat up our rooms. Warm air rises, its place taken up by the cold air, so it warms up in circulation till the whole air is warmed up. And that is called convection. Convection. Then the last one is known as radiation. So radiation, that's heat transfer. The heat transfer is concerned with exchange of thermal radiation between two or more bodies. Thermal radiation is defined as electromagnetic radiation in the wavelength range of 0.1 to 100 microns, very tiny wavelengths, which encompasses the visible light regime and arises as a result of temperature difference between two bodies. So here, no medium need to exist between the two bodies for the heat transfer to occur. So in radiation, you don't need any contact anywhere, like the sun. Take the sun, energy from the sun, and then somebody standing, you don't need to have contact. You can still feel the radiation from the sun. It's a radiation. So you don't need any medium in between. It can be vacuum. You can still travel between the vacuum and get to the object. Okay. And all these are electromagnetic waves. Electromagnetic waves. Okay. Electromagnetic waves don't need any medium to travel through. Okay. Radiation. So we feel the sun's heat by means of radiation. They release electromagnetic radiations, very small wavelengths. So those are the three types of heat transfer. Okay. So if you think about the thermal flask, the flask is created in such a way that it reduces conduction and radiation. Conduction, radiation to reduce the heat loss. Okay. So a simple flask will have like two walls. If you take the flask, the inner side is shiny. It's like a two-layered flask. It looks like this, and they have the outer covering. The base is an insulator. So let's say this is a flask. Okay, so you see that the outer, you have a double layer here. Okay, and this shiny surface, the inner, you have the shiny surface. This is shiny. Okay, and there's air in here, in between those shiny surfaces. Now, air is a back conductor of heat, right? So that's why I have air here. The shiny surface reflects. Shiny surfaces, they reflect heat. So it does not reflect the heat inside, you know. And then the base here is an insulator. So the insulator, if you put this on the table, is going to reduce conduction, loss of heat by conduction over here. Okay. So that's how the flask is made, to reduce heat loss by these methods. Okay. All right. So know about the different types. And then the other part is to know about calculations. Calculations involving quantity of heat. How to calculate quantity of heat. So I have an example here. Okay. To calculate the quantity of heat, I want you to remember this formula. I'm going to memorize this formula to use. Okay, I'm going to break it down for you to understand. So the quantity of heat gained or lost by a substance is equal to the mass of the substance, that's M, multiplied by what you call its specific heat capacity. Specific heat capacity, I'll explain that to you. Multiplied by the change in the temperature. So the change in temperature is initial temperature, final temperature minus the initial temperature. You know, that's what we made a change in temperature. So in some books, we see maybe Q is equal to MC delta T. Delta T may change in temperature. Change, and that's final minus initial temperatures. Okay. That is what we refer to as quantity of heat. Okay. So what is specific heat capacity? The specific heat capacity is of a substance is the amount of heat required to raise the temperature of 1 gram of a substance by 1 degree Celsius. So you have 1 gram of a substance, how much energy, heat energy do you need to increase this temperature by just 1 degree? That's what it means by specific heat capacity. So every substance has its own heat capacity. That for water is different, that for metal is different, so on. You know, for water, usually it's like 1 gram per degree Celsius. If you have water, it's like 1. Okay. Then you have heat capacity. Heat capacity, know the difference. Heat capacity is defined as the mass of a substance multiplied by the specific heat capacity. So if I multiply heat capacity by the mass, it gives me what we call the, if you multiply the mass by specific heat capacity, then you have the heat capacity. That's what we call the heat capacity. In other words, you're looking at this part here, MC. This MC is the heat capacity. That's the part we call the heat capacity. So we can say the quantity of heat is the heat capacity times the change in temperature. Okay, I have that here. That's what I have down here. Okay. So how do you use this in calculation? Let's look at a question. So this example says, calculate the quantity of heat needed to raise the temperature of 250 grams of water from 20 to 56 degrees. 20 to 56 degrees. Okay. And then they gave us the heat capacity of water as 4.1 grams. Okay. So quickly, I would ask myself, what is the mass? The mass is given. Actually, this one should read specific heat capacity. What I have, the C here. This here should be, let's add specific heat capacity to this. Specific heat capacity, not just the heat capacity, specific for this one. Okay. All right, so you're going to write down your formula. Q is equal to MC times the change in temperature, right? You know the mass is given. The temperature is given. The initial temperature, final, initial. So write all those things down. So there's a mass, final temperature, final, and this is the initial temperature. So the change in temperature is going to be 56 minus the 20, which is 36. So 250 times the specific capacity times the change in temperature. If you work this out, then you get the quantity of heat to be 37620, right? Kilojoules, the unit is joules. The unit will be in joules, the heat energy. Unit of heat energy is joules. Okay. But sometimes they give you the answer in kilojoules. Kilojoules is a higher unit. So we know that 1,000 joules, J is joules, right? Is equal to 1 kilojoule, kJ. So 1 kilojoule. So that's a conversion factor. So here, if you convert this, then you get, it means you have to divide by 1,000. And if you divide it by 1,000, you have 37.62 kilojoules. If you round up, they have about 38 kilojoules here. That's where that answer came from, rounded, to the nearest tens. So that's the formula that you need to use here. So these are some questions that you can practice on yourself, based on what I have there. I have the answers already. So I'll leave that to you so you can practice yourself. Okay. So calculate the amount of heat needed to increase the temperature. This is the one that we did. Okay. Then B, calculate the specific heat capacity of copper, given that 204.7 joules of energy raises the temperature of 15 grams of copper from 25 to 60 degrees, right? So if you list everything, you'll be given the heat capacity that is given, that's the heat capacity. You know the mass, you know the initial temperature and the final temperature. So if you use a formula, you have the heat capacity is given, the amount of heat is given. So this is the amount of heat given. That's this, your Q is given. You know the mass, this is a mass. Specific heat capacity we don't know, so you C to represent, or maybe X or something, a variable. And then a change in temperature is 60 minus that. So if you put this in and you work out C, you should come up with this answer. Okay. So you should be able to try that yourself and come up with this answer. So it's all about the formula. You put in the information given and you find a missing part of the question. Okay. All right. So that is one aspect of heat capacity, of heat. You should be able to calculate the quantity of heat, or maybe find the final temperature, or find the heat capacity and so on. Yeah. So play around with that yourself. Okay. All right, then the other aspect is to know what we call change of state. Change of state. Okay, change of state. What do you mean by change of state? It means that you are changing a substance from maybe a solid to a liquid, or liquid to solid, and so on. Okay, remember the 3 states of matter: gas, liquid, solid. You should know the properties of each one. Solids have fixed shape and fixed size, right? I hope you remember those parts. Liquids have what? Fixed. They are fixed volume, right? But they don't have fixed shape. And then gases have no fixed shape and no fixed size. You should know those properties. So the change of state, if I change, let's say, gas directly to solid, we refer to that as resublimation. If I change solid directly to gas, then we call that sublimation. So it comes up on the test sometimes, sublimation. It means I'm changing a solid directly to gas, without going through the liquid. One typical example would be if you take an example that they give is dry ice. Dry ice, okay. Dry ice is solid. If you leave it for some time, it vanishes, becomes a gas. It doesn't become liquid, right? Just change the gas. So that's sublimation. Dry ice, the name for dry, this is carbon dioxide. It's made of carbon dioxide, the dry ice. They're talking a lot about it these days for the vaccine, to store the vaccines in dry ice. Keeps it cold. So that's sublimation. Another example I can think of is like the mothball. The mothball. The mothball, the one that you can use it to drive away insects and so on, put in the corner of your room, and then drives away rodents. If you put it down, it's white and solid, right? If you put it down, it doesn't become liquid. After some time, it vanishes, it vaporizes, it becomes vapor or gas. That is an example of sublimation. Another example would be if you take the, what do you call this thing? Air freshener. You have the solid one, the solid air freshener. You see that you leave it, it's like jelly, put it down for some time, it doesn't become liquid, it just changes to gas. These are all examples of sublimation. So you can have all these examples and they ask you to identify what change of state you are dealing with. Okay. Now, if you change liquid to gas, then you have vaporization, right? Or vapor, you're boiling something of vaporization. Gas becoming liquid, then that's condensation, condensation. If you have liquid becoming solid, then you have freezing. You have freezing it. And you have solid becoming liquid, melting. So those are the change of states we're talking about. Okay. Now, this diagram is known as a change of state energy diagram. Okay, we'll see one in your book when we start looking at the questions. You have to know this diagram very well. That's why I put it here. Now,
I have a question.
Yes, please go ahead.
Hi, sorry. Can you give me a direct sample of resublimation? The only thing I can think of is gas turning into
It's
liquid.
right here. Sublimation.
No, resublimation. Gas direct to solid.
Yeah, if you have a gas, if you have a gas that you change to solid, you condense it to a solid, it's what we call resublimation.
Is there a direct sample for that one? The only thing I can think of is gas turning into liquid and liquid turning into solid, like evaporation, I guess. Or like when a gas. Oh, wait,
Yeah,
no.
so I mean, you can have carbon dioxide.
Condensation.
You have carbon dioxide that you can resublime it to gas, to solid, to form a dry ice. Carbon dioxide is it's a gas.
Oh, when you make it. All right, cool. Thank you.
Right. Yeah. So if you, it can cause it to become solid, becomes a dry ice. That's what we call a dry ice. Right. So that dry ice that you see is carbon dioxide. We know carbon dioxide like a gas, which has been made into a solid form. Okay. Yes.
Right, thank you.
You're welcome.
Thank you.
Yes. Okay. So this diagram here is called the energy diagram. Okay. We'll see one in your book shortly. It looks like this. So you have temperature on the y-axis and then time on the x-axis, like this. It can be changed. But if you understand what I'm going to talk about, even if they switch the labels, you should be able to make it out. So let's look at this carefully. I'm going to start with, let's say, have water, ice at -20. We start at -20 degrees Celsius. It's Celsius here. So I'm going to draw ice. The temperature is -20 degrees Celsius. Okay. Now this ice, let's say that I want to change this to, I want it to melt, right? Put in heat. Let's put in some heat. So if we put in heat, what's going to happen? The temperature is going to increase. That is what you're seeing here. I'm going to use a highlighter. The temperature is going to increase. So from -20 to about 0, it's going to get to 0. Okay, so this is what is going to happen first. The ice will increase to a temperature of 0 degrees Celsius when I put in heat. Then I keep putting heat. Let's keep more heat. We're putting more heat. What happens? You're going to realize that the temperature will stay at 0 for some time, even though it's still a liquid. So ice and a liquid will be at what we call equilibrium. So ice and liquid in the liquid and ice equilibrium. So it changes to liquid, the temperature will still be at 0 degrees for some time. That's what you see here. The melting. So at this point here, we have solid and liquid at equilibrium here. The changing, you're putting heat. It appears as if nothing is happening here. That's why it still stays at 0. Where is all the heat that you're putting in going? When you measure the temperature, it's still at 0. You are putting in heat, but it's still 0. Why? That's the question. It's all because there's what we call a change of state at this here, the flat line here. Change of states. There's a change of states here. Okay, so it stays at the same temperature for some time. Now, the energy that you put in is used to break the bonds. It breaks the bonds in the ice, the solid state, to free them into the liquid state. Okay, so the energy here is known as the latent heat of, we call it fusion. So latent heat of fusion. Latent means like hidden, apparent, hidden, So something's happening there, but you don't see it when you measure a thermometer. Because the energy is used to break bonds, breaking bonds in a solid state, so the molecules can now move freely, in the liquid phase. Okay, latent heat. So important to know latent heat. So there's a change of state here. So what we are dealing with here is the latent heat at this state, latent heat of fusion. That's what's occurring here. Then once the molecules are the bonds are broken down to molecules, and now you have the liquid phase. As you put in heat, now putting more heat, what happens? The temperature is going to increase rapidly. So that's what is happening from here. You're in the liquid phase now, temperature shoots up and gets to about 100 degrees. So you still have a liquid here. You're going to get to the liquid at 100 degrees, which is water at 100 degrees. Or let's say water, 100 degrees. It shoots up to 100. Then keep adding more heat. As you keep adding more heat, you're going to realize that now the temperature stays the same when it's changing to it's about to change to a gas phase. The temperature will stay the same. So gas, 100 degrees for some time. So the liquid is in equilibrium with the gas. At the same time, it's changing, so it's equilibrium over there. So temperature remains 100 for some time, and that's what is reflected here. As time goes on, it stays for 100 for some time. Why? Because again, there's a change of state here. It's a change of state. It's changing from liquid to gas, change of state. So the energy is used to free the molecules, break the bonds between molecules in the liquid so that it freed them into the gas phase, right? So again, there's no change in temperature here, and the heat that is energy over here is known as the latent heat of vaporization. Vaporization. Energy is used to break the bonds to free the molecules in the vapor phase. Then once you free them into a vapor phase at 100 degrees Celsius, keep heating up, and then the temperature will shoot up. The temperature can now increase, you know. So here now I can keep going, increase to a higher temperature after 100. Gas above 100, more than 100 degrees. You know, that's what happens. So this is the diagram you should know very well. Change of state occurring where the change of state, okay, flat line, same temperature, not changing at those points. There's a change of state. So it is a vaporization, heat of latent heat of vaporization here. Okay, change of state. So we see this diagram, you should know what's happening. Okay. So as I said, okay, the same thing I have here, you know, more illustrations. You know, phase, change of phase right here. You know. Yeah, so even if this diagram is turned upside down, the other way around, you should be able to tell what's happening. Okay. Sometimes they can draw the diagram that would look like so instead of I can put the time here, I can put temperature here. You should be able to tell me the same thing. So the temperature is here, let's say -20 here. And then let's say we have 0 there. Let me put 100 here. So we can have, as time goes on, the temperature will increase to 0. And as time goes, it can stay at 0 for some time. This is at 0. And then it increases. And then it's going to stay at 100 for some time. If you see something like this, the same thing. So it didn't change at 0, same temperature here, 100 as time goes on. So this will be a change of state here. So the latent heat will be here. Latent heat is of fusion. So there's a change of state here at this level. And then there's a change of state here as well. So this part here is going to be your latent heat of vaporization. You know. So no matter how they turn the diagram, if you know what's happening, you can always say what's going on. Okay. All right. So any question on the heat diagram? Same temperature not changing. That's the way you can remember. Where there's a no temperature staying the same for some time, there's a change of state there. Okay. And then you have another type of diagram that they also ask you. If you've gone through the book, you may have seen this. It's called a phase diagram, the phase diagram. Now, the phase diagram shows you the relationship between pressure and temperature, you know, and what happens to a particular substance. So let's say if you take water in the lab, you change the temperature, you change the variable pressure, and then you see what's happening. If they do that, they come up with this diagram that looks like what you have here. You know, you have like a line, like a Y-shaped type of graph when I face that. So this line here, this represents this like a solid, the line for solid. This will be like the line for like liquid. Solid area is here, and then vapor area is here, you know, something like this. Okay. So what I'm talking about, it looks like this. Okay. So this is a solid area. You have the water liquid area. Anything here is liquid. And then here maybe like a vapor or gas region. Wherever the three meet this point is known as a triple point for that substance. This is called a triple point. Okay, so at the triple point, it's the point, the temperature. So this is temperature here, temperature on this side, and pressure. The triple point is the temperature at which all the three phases, liquid, solid, they all occur. It's like they are at equilibrium. It's like an equilibrium point for all the three phases, equilibrium. In other words, if you take that temperature, you have a liquid, solid, and gas all existing at the same time, you know. And that's how they came out the temperature 273 Kelvin for water. You know, is a temperature where you have all the different states of water occurring. Triple points. And that's why we have triple point here. Now, what are the questions that they ask you here? The type of question that they ask you is this. Usually they can do very straightforward questions. Yeah, you just have to know what they're talking about. Let's say A, then they have an arrow maybe to B. Or I can have, let's say, C to D, and maybe E to F. Okay. Then a question like, which of the following represents maybe sublimation, right? Sublimation. So quickly, you know that sublimation is changing from solid to gas, right? Solid to gas. So which one is changing from solid to gas? It's EF, right? If you look at EF here, if on solid state, the solid state and going to a gas phase. So EF represents sublimation, you know, something like that. EF. This is sublimation. EF sublimation. And if you take like AB solid to liquid, AB from solid to liquid. So since it's moving from solid to liquid, let me change this a little bit here. Okay, so you have this moving from solid to liquid. So solid, that's melting. So it's melting right there, and so on. You know, C to D, liquid to gas. C moving to D is from liquid to gas. So if something's moving from liquid to gas, then we know it's vaporizing, so evaporation. Evaporation. And so on. That's how you can use this diagram when they give to you. All right, so the phase diagram, the answers are in the question from the diagram. Okay. Okay. Great. Any more questions on this? Any other questions?
No.
Okay. Yeah, it's going to be very simple. Once you go to the book, you see how fast you can move with all the knowledge I'm giving to you. Okay. All right. So I think the last part we're going to look at today is genetics. Even once I do, we've done very well. We've covered a lot. So genetics will be the last topic under the science. And then we'll do math. I want us to do a lot of math today. Okay. So far we've done very well with the science. I've covered almost everything I need to do for today. Okay. All right. So let's look at genetics and then we'll practice. Okay. So genetics, we've already talked about RNA and DNA, the differences on day one. So I'll skip the intro here and go straight to the genetics. So things I have to know to answer genetic questions. We have to know what a gene is. The gene is the unit of heredity. Okay, we know that. A section of DNA sequence encoding a single protein. Then the new thing you have to know is alleles, when they're alleles. So alleles are two genes that occupy the same position on what we call homologous chromosomes and cover the same traits. So in other words, a pair of genes that control two contrasting characters. That's another definition. Okay, two genes controlling two contrasting characters. Example, let's take something for height. If we take height, there are two genes that control height, one for tall and one for short. Okay, for tall, we use the letter uppercase T, and then short we use the lowercase t. And I'm going to explain what this is. The uppercase means that it's called a dominant gene, and lowercase is known as the recessive gene. We'll talk about that. Recessive, dominant, and then a recessive genes. Okay. So these two genes are known as the alleles. So T and T for height. If we use height as an example, the pair of genes controlling two contrasting characters, height, short, and tall. They occupy the same position on the chromosome, okay, and cover the same trait, heights. Then the locus is a fixed location on a strand of DNA where the gene or the allele can be located. Where you find a gene on the DNA, that position is called a locus. Then we need the word homozygous. The key words I'm highlighting, very important, homozygous. So homozygous, they have identical genes, one from each parent for a particular characteristic, homo same, homozygous. So for example, if the person has TT, two genes, TT uppercase, or two lowercase, two recessive genes, then these are homozygous individuals. So in the question, if they mention homozygous, they know what you're dealing with, same genes from both parents. Then heterozygous, hetero, means different, two different genes. Okay, example, let's say uppercase and lowercase t, then this is a heterozygous individual. And I mentioned a dominant gene. Dominant gene is a gene that is a trait that appears in the heterozygous condition. So when you have heterozygous conditions like T, uppercase T and lowercase t, the dominant one will always express itself. So like height, tallness is dominant. So it always shows itself. It will suppress the shortness. So that is a dominant gene. And a recessive gene, so you should know this one, what it means in a question. Recessive gene, the one that is masked, it doesn't show itself. So like this T, uppercase T and lowercase t is going to show, the T is marked, so that's the recessive gene. That's the definition. This T is suppressed by the uppercase. Okay. They have a phenotype. So phenotype is what is the outward expression, what you see on the outside, or how the person expresses. So if I have something like uppercase T and lowercase t, because T is dominant, uppercase tall is dominant, this person is going to be tall. Okay, you're going to show tallness on the outside. While somebody who is this is also going to be tall. And somebody with lowercase t's, this person is going to be short. That's outward expression, what you see on the outside. Then genotype, what makes up the genes, the person's genes, genotype. Genetic makeup is a genotype. So they ask you genotype, genetic makeup. And genetic makeup, let's say the person is uppercase homozygotes, or maybe heterozygotes, or maybe homozygote for recessive, like this. That's a genetic makeup. And then based on this, they can ask you the ratios. What are the phenotypic ratio? What are the genotypic ratio? And so on. You know, how do they show what the percentages? So that's what we're talking about here. You know, so those are some keywords to look out for. And we have what we call Mendel's laws of genetics, inheritance. Mendel, he did a lot of work on genetics. The first law, Mendel's law of dominance, it says that when an organism has two different alleles for a trait, one allele dominates. Okay, so one is dominant, one dominates. And second, that's law of dominance. And the second law, Mendel's law of segregation. Segregation, like separating. So it said during gamete formation by a diploid organism, a pair of alleles for a particular trait separate or segregate during the formation of the gametes, as in meiosis. Okay, meiosis. So in other words, when you have, I'm going to do what we call the Punnett square for you to see later. So they separate randomly. Okay, I mean, they can go into different sperms or eggs, the genes. So if the person is T, and let's say this, the one parent has that, and that parent has, let's say, lowercase. Okay. So random, so segregation means that this separates. One egg can have that, the other egg, the sperm can have this, you know, randomly, you know, independent of each other. They can go to different egg and sperm. Then when we talk of the law of independent assortment, so the members of a gene pair separate from one another, independent of the other. These separations occur in the formation of gametes during meiosis, right? So the random assortment, that means that the members, they can separate from one another. So this can combine with this. That's what it's saying, right? Now, this can go with different combinations of each other. This can combine randomly with, and so on. Okay, assortments. Okay, so those are his laws. So let's look at an example. I'll make up an example here. Let's say that you have homozygous female, right? So you have a homozygous female, for tallness. Maybe marries a man who is heterozygote. For tallness. What is the genotypic ratio? Genotypic and phenotypic ratio. Ratios. So here we should say, since I'm looking for the two, let's make it what are the genotypic and phenotypic ratios? Okay, this question like this. So the keywords are the homozygous female. So you have to identify homozygous female, identify the homozygous female for tallness, and then identify that for the male. Okay. So I'm going to use what we call the Punnett square. The Punnett square for this. So the Punnett square, you do something like this. And here, let's put a male here. So usually they'll do something like that, like male, and maybe female here. A female like this. So since the person is homozygous, if the person is homozygous for tallness, we know tallness is dominant. So it means that the gene for this person is uppercase T's. So the female, so we're going to put one in a box here and now in a box there. So that's why you have the segregation, okay, separating. Then for the male, the genes will be heterozygous. So it means it has uppercase for tallness. So in the lowercase, two different genes. So it's like separating them into different gametes. Now we're going to do the random combination here. So T uppercase can go with that. So I have uppercase this and that. This can go with this. This can match with that. This can fertilize that. So these are all the possibilities that you have, the different combinations. So when you look at this, now you're going to answer the genotypic ratio. So let's go at genotype, genotypic ratios. So that what genes are they made up of? So we are looking at these here. So we have 4 possibilities in all. Okay, 4 possibilities. So here we have 2 out of 4, the ratio. So it's 2 out of 4 T uppercase homozygous for tallness. And then we have 2 out of another 4, that T uppercase and lowercase. So another 2 out of 4 uppercase. So these are heterozygotes. Okay. In other words, you have 50% or half of this, and then 50% of that. Okay. Or someone can say the ratio is 2 to 2, which is 1 to 1. 1 to 1 if you reduce to lower stems. 2 to 2. In other words, let me write everything out. It's like having 2 TT, 2 to that, which is 1 to 1 ratio for this. 1 to 1 ratio. So that's the genotype, the genes that they have. Now, what about the phenotype, phenotypic ratio? The phenotypic ratio will be what you see on the outside. What do we see on the outside? On the outside, we have this person here, TT. This will be tall, this person will be tall, this person will be tall, tall, because T is dominant. So the phenotypic ratio will have all tall. We see that they're all going to be tall. On the outside, they're all tall. So 100% tall. Okay, that will be the ratio here. Okay, using the Punnett square. Okay, so that's what we have. So they ask you questions like this, that you should, what are the ratios? The other way they ask the question, they work backwards. They give you the ratio of the offsprings, the offspring are like the children. And then they ask you what are the parents, the genes of the parents. In those cases, we can work backwards from the answer. You take each option and then do the Punnett square and find out the ratios, till you get what matches. So it's like backwards. Okay. So Punnett square, you should be able to draw the Punnett square easily when they give you a question to answer. Okay. And then there are special cases, some special cases that you come across known as the incomplete dominance. Incomplete dominance, in that case, the gene doesn't dominate completely. Doesn't dominate completely. Like example would be like red color. If you have white flower and then some that are red, sometimes because of incomplete dominance, you have the offspring, if you cross the two, it becomes like pink. Because the red is not completely dominant over the white in those type of flowers, so it becomes pink color. If you see that, you're dealing with incomplete dominance. Okay, it's not fully red, not fully white. And then you have something called multiple alleles. Certain conditions, you have more than one allele controlling the same type of trait. For example, the black group. If you take a black group, it's determined by a single gene with 3 possible alleles. 3 possible alleles for the gene. It can be A, B, O. So those A, B, you have A, you have black group A, black group B, black group A, B, and black group O. They are all controlled by the genes A, B, O. Different genes that gives you different, for the same trait, but have different possibilities. They have multiple alleles. Okay. And there are genes that we say that they are co-dominant. So A and B, they are co-dominant. So somebody has black group A and black group B, the person doesn't become like A or B, but the person is AB. So you know that they are co-dominant. None of them is dominant over the other. So you have called a co-dominant. They're equally powerful. Equally powerful. They are both expressed the same. Both alleles are expressed. They will say they are co-dominant. You have A, they'll have the A gene and the antigen, and they'll have the B antigen, at the same time. Both are expressed. Okay, so that's what we mean by this. And then the other part is called the sex-linked inheritance. Sex-linked. Sex-linked inheritance, you see we have 23 pairs of chromosomes in the human cell. 1 pair is known as sex chromosome, and the remaining 22 pairs are referred to as the autosomes. The autosomes. Okay. So the sex chromosomes determine the sex of the human. There are 2 types, X and Y chromosomes. Female have XX, and the males 2XX, and then the males have 1X and 1Y. Okay, so we have XY for males. In humans, the Y chromosome is much shorter than the X chromosome because this is short. Because of its shortened size, a number of sex-linked conditions can occur. When a gene occurs on the X chromosome, the other gene of the pair probably occurs on the other chromosome. Therefore, the female usually has 2 genes for a characteristics. Okay. So in the male, we can have certain conditions which are not seen in females because there's no other, there's no other gene to balance it or counteract it on the Y chromosome, and that causes a lot of diseases. These are some examples of sex-linked inheritance. If you take color blindness, is an example. The gene for color blindness is found on the X chromosome. So a woman is rarely colorblind because she usually has a dominant gene for the normal vision on the other chromosome. However, a male has a shortened Y chromosome. Therefore, he has no gene to offset a gene for color blindness on the X chromosome. As a result, the gene for the color blindness expresses itself in the male. Okay, so that's an example. Another example is hemophilia. Hemophilia is another example that runs through the royal family and so on. So in hemophilia, the blood does not clot normally because an important blood clotting protein is missing. The gene for hemophilia occurs on the X chromosome. As females have 2 X chromosomes, 1 X chromosome usually has the gene for normal blood clotting. Therefore, the female may be a carrier of the hemophilia, but normally does not express hemophilia. Males have no offsetting gene on a Y chromosome. So the gene for hemophilia expresses itself in the male. This is why most cases of hemophilia occur in males. Okay. So when they give you X-linked inheritance, you use X and Y chromosomes. I'm going to do one for you to see. So when I say X-linked inheritance, let's say that you have a female who is a carrier. So female carrier. Carrier means has a gene, but doesn't have the disease. You know, so female carrier for hemophilia. So let's say this one, hemophilia. Okay. So the person is carrier, then you have, remember the first XX. Hemophilia, this is a recessive condition. So I'm going to use H, lowercase h, uppercase H for hemophilia, right? So the person is a carrier, means has a normal gene, and then one of the genes abnormal. So the H, we're using the H is the abnormal gene, abnormal. Okay. And hemophilia is a recessive disease. Okay. So that's why we use lowercase for this. Abnormal gene, recessive. So that's a carrier. And then if let's say I have a male, maybe the person marries female carrier. Okay, so let's say person marries a male, and the male is, let's say this person is normal. Can marry a male who is normal. What will be the offspring? Okay, so let's do that and see. So I'll do a Punnett square. So let's have the female here, and then the male here. Or I'll put the male on top and the female, and the male, this is a male, and then put a female up here. So the female is a carrier, so carries a gene, but normal. So X, H normal here, then another one will go abnormal. Since this person is normal, it's going to have X, H, X, no, and Y, because the male is XY. There'll be nothing on the Y. So this will be the gene for the male. So carrier normal means that it's X, H, Y. Person is a male. Okay. So what happened when you do the crossing? This will interact with that. So fertilizes this, fertilize that. This X here, normal, abnormal, this and that XH with a Y, and then this will be X, H, and Y. So what is happening here? Let's look at that. So we're looking at what is happening, the offsprings. Okay. So first, when you're analyzing this, look at the males and look at the females. You're going to look at the different genders, okay? So among the females, what's happening? So the let's call it phenotypes. Or phenotypic ratios. Now, amongst the females, so these are the females. These two are the females, this and that. What do we see? We see that we have half-half. 1 out of 2 normal, and the other one is carrier, right? So, but they're all normal because this person will not have the disease. So we're going to have all of them normal. Females are normal. Females, they will not have the disease, no hemophilia. They are all fine. Okay, but half of them will be carriers. You know, another here we have half, so 50% if you look at the 50% carriers. Half of them will be carriers. That's all, but they don't have the disease. They just have the gene. Then what are the males? If you see the males, phenotype, if you look at the males, this and that, half of them normal, this is normal, and this is will be abnormal, be affected. So we see that 50% normal, and you have 50% who are having the disease. 50% have hemophilia. They have the hemophilia. Okay. So 50-50, that's what you see here. Okay, so that's how you analyze, you look at the genders, and then you play around with that when in a sex-linked inheritance. Okay. Okay. Any questions so far on this? Okay. All right, so I'll pause on this here. All right, so now let's take a book and then we go through sample questions for the next few minutes. So first question, page 218. 218. Number 5, we know the answer. The alimentary canal is associated with A, spinal cord, B, digestive system, C, urinary tract, and D, birth canal.
B.
B.
B, correct, digestive system. Okay. Number
What is
6.
the page number again?
218. 218. Okay. All right, the next page is 219. Number 6. The numerous villi in the small intestine serve to A, secrete enzymes for digestion, B, absorb water from dissolved food, C, secrete hydrochloric acid to dissolve food, and D, provide greater surface area for absorption.
Absorb.
Yeah, so D provides a greater surface area for absorption. Okay, we saw that. Okay, good. All right, now let's look at page 221. Page 221. So that has to do with one of the energy diagrams, number 17 and 18. So look at that and then tell me what you think the answer will be. Number 17 and 18. Okay, so 17 says the number of calories needed to change the liquid to steam at its boiling point temperature is, so changing from liquid to steam at the boiling point temperature.
Is it A?
It's A, correct. It's A. So in that diagram, you're looking at 100 degrees, and in the 100 degrees, it's just like the temperature didn't change at that point. So there's a change of state at that point, and it's 540 calories as indicated there, correct. I hope everybody's good with that question. Anybody confused with that question? So that's
Yes, can you explain that?
okay. Look at 100 degrees. You see 100 degrees? That's
Yes.
where the change is changing from liquid to steam. vapor, liquid to vapor. So you have 100 at that point, 100 degrees. That's a boiling point.
Yep.
All right. So they're asking you what's the number of calories. And you see 540 calories. It's written there.
Oh, I see. Thank you.
So yeah, that's the energy that you need. Okay. Okay, let me change the question around. So what will be the calories needed to change from solid, like from solid to ice to liquid? What will be the calories on the same diagram? Ice to liquid or water, liquid water.
Is it
How many?
80 calories?
It's 80, correct. It's 80 calories. Because if you look at a change of state, 0 degrees. That's why you're having ice changing from ice to liquid, 80 degrees. The temperature stays at 0 for some time. It's a change of state over there. So in that case, we 80, correct. Okay. All right, so number 18. If the temperature were raised to 300 degrees Celsius, the substance would A, change back to liquid, B, remain a gas, C, condense, and D, break into its constituent elements.
Is it B? Remain a gas?
Yes, it will remain a gas. It will remain a gas. All right, so after, if you look at 300, it's above 200 in the diagram. So it's a gas phase that's changed to a gas. It's still in the gas region. It's still a vapor. So to remain a gas, correct. Okay, so that's one way in which you ask questions on the energy diagram. Okay, page 223. Page 223. Number 29 through 31. So this is a diagram on the endocrine system that I was talking about. So number 29, it says the function common to the organs of this system is A, reproduction, B, secretion, C, respiration, and D, excretion. So which function is common to all the organs?
Secretion.
Yes, secretion. So releasing hormones directly into your blood. Secretion. Okay. So number 30. Which of these organs controls the amount of calcium in the blood? Controls calcium in the blood. A, 1, B, 2, C, 3, and D, 6. So which part's labeled amount of calcium?
Six. D.
6 is the adrenal gland. So 6 is the adrenal gland, so that doesn't control the calcium.
3.
Yeah, 3. 3. So 3 is pointing
Parathyroid?
to the parathyroid. It's pointing to the parathyroid gland. Okay, on the 3. So that controls the calcium level. Parathyroid would increase the calcium level in the blood. Okay. Calcitonin will reduce, right? Those are but the diagram is pointing to parathyroid. Okay, number 31. Which of these organs has the added function of producing digestive enzymes? added function of producing digestive enzymes?
7. D.
Right, so let's see, 7, that's correct. So 7 is the pancreas. So pancreas, both exocrine and it's endocrine. So it has added function. So that is correct. Good job. All right, so you see how some of the questions go in this. Okay, another one. Let's look at page 227. Page 227. Number 51. Number 51.
C.
Okay, says the boiling point increases by 20. Yes, you are correct. You're correct. So the pressure on water is increased from 1 atmosphere to 2 atmospheres. How, if at all, is the boiling point affected? So if you look at the 2 atmospheres, you see that the temperature is 100. Sorry, 1 atmosphere is 100, and 2 atmospheres is 120. The point D. So the difference is 20. Okay, that's correct. So that's one way, the phase diagram, you can get a question. All right, page 228. Page 228. Number 58. The vaporization of solid dry ice to gaseous carbon dioxide by heating is an example of?
Sublimation. A.
Yeah, this is an example of sublimation, right? So we can talk about resublimation here as well, so sublimation. So that's one example. Okay, page 229. Number 1. The organ in the human female that develops to nourish the embryo is called A, amnion, B, yolk sac, C, fallopian tube, and D, placenta.
Placenta.
Placenta, correct, because nourishment, so placenta. Amnion is a covering around the, protective covering around the embryo. Placenta nourishes. Okay, what about number 3? What will be the likely result if all the bees and butterflies in an area were destroyed? Likely results if you destroy all the bees and butterflies in an area.
D.
All
Fewer
right, so,
plants. Fewer plant seeds.
yeah, fewer plants seeds will be produced. Correct. Okay, fewer plants will be produced. Remember, I said no fertilization, no pollination, no seed production. That's where your mind should go to. Correct. So you have fewer seed produced. Number 4. The tiny projections in the small intestines adapted for absorption are called A,
Villi.
venules, B, villi, C, alveoli, and D, nephrons.
B. Villi.
Villi, correct. Villi. So that's good. All right, so another question. Page 232. Number 13. 232, number 13. Most of the work done by the human kidney occurs in A, nephron, B, neuron, C, ureter, D, alveolus.
A.
A, correct, nephron.
A.
Remember, we said that's a functional unit of the kidney, nephron. Okay, correct. Let's look at number 19. Number 19. So it's a diagram that has to do with the heat thing that we discussed, some of the heat problems. So you have 4 test tubes are filled with water and placed in a test tube rag, as shown in the following diagram. 3 of them are covered, each with a different material. So the first one, you have a black paper, second white paper, third one aluminum foil, and a third no cover. And have thermometers in them. So it says a thermometer is placed in each test tube, and the test tubes are placed in bright light for 15 minutes. In which test tube would the temperature rise most rapidly? They have A,
C.
B, C, D.
C.
You said C. Okay,
See
what's
the
your reason?
radiation. The heat transfer, the change. Hold on. I got this.
You got it?
Yes, it's something to do with the radiation, like the heat transfer.
Mm-hmm.
So there's no direct medium to be used. So the sun was directly heating the aluminum foil.
Okay, but you see, the aluminum foil is going to reflect.
Oh, that's
It
right.
will reflect the radiation.
That's true.
Yeah, it's like almost like the way the mirror reflects. So you're going to reflect. It's a reflector.
So this is what, a convection then?
Well, this is going to be like radiation since it's the sun. The
Yeah,
sun
but
is hitting
but
it,
it-
so radiation.
okay.
Yeah, but look at it. Think of everyday life. Why do we put on white and sometimes put on black if they like if it's cold?
Okay.
You know, black and there's something has to do with everyday life. Think about it like
The
that.
sun absorbs.
Yeah, absorbs. So see, if you put on black when it's sunny, you're absorbing the heat, right? So you warm up, faster. White will reflect, like white substances tend to reflect the heat. That's why I want to put something that's whitish, to reflect. Okay, but black, if you put on black, it absorbs. Feel hot. You're gonna feel hot. Okay. So it's a black one that will absorb. The white paper, aluminum foil, they reflect the light. No cover, yes, it's gonna absorb the sunlight, but then you have a black one. The black paper will absorb more. Black objects tend to absorb more heat. So A will rise faster. That's the reason. Yeah. So A should be the correct answer. Now, number 20. It says, in the diagram of the bean seed, which letter represents stored food for the growing embryo? So you see the diagram of the seed diagram. So
Is
which
it
one?
A?
Yeah,
A.
A, correct. A, the bulk of the seed. A, that represents stored food. That's correct. A. That's correct. Okay. So those are good examples here. Let's see another example. Okay, page 237. 237. Number 35. 237, number 35. Say in pea plants, plant height and seed color obey the law of independent assortment. If seeds are hybrid for both of these traits, how will their offspring most likely appear? A, all tall with green seed. B, all short, some with yellow and some with green seeds. C, some tall, some short with all green seeds. D, some tall and some short with either green or yellow seeds. So there's a keyword that helps you get the answer.
Is it D?
D, some will be short, some tall, some short, either green or, yes, is correct. So it's a hybrid of traits. Okay, so some will be tall and some will be short with either green or yellow seeds. Hybrid, mixture of different traits. Correct, D. Okay, page 238. Page 238. number 38. A student studies a structure microscopically and knows that it contains glandular cells. It has blood vessels entering and leaving it, but it has no duct leaving out of it. The structure must be A, an endocrine gland, B, an excretory gland, C, a digestive gland, and D, a tear gland.
An endocrine gland.
Yeah, so it's an endocrine gland, ductless, no duct. That's the key word. And a lot of blood vessels entering, leaving it, correct. All right, that's good. So far, okay, let's go to see here. Okay, page 245. 245. Number 19. Which of the following structures is a functional unit of the human lung? A, bronchus, B, Malpighian tubule, C, nephron, and D, alveolus.
Nephron.
Nephron is for the kidney.
D, alveolus.
D, alveolus. Alveolus. So for the lung, the functional unit is the alveolus. Alveolus. Okay. All right, look at number 21. 21. If a person's gallbladder were removed by surgery, which of the following substances would he have the most difficulty digesting? A, carbohydrates, B, nucleic acids, C, fat, and D, proteins.
C,
C,
fats.
fat.
Yeah, C, fat. Remember the gallbladder stores the bile, which helps to emulsify fat. So that is, you're going to have difficulty digesting fats, correct. Yeah. Okay, let's just add 22 to this one. So which of the following phenomenon is most likely to speed the evolutionary rate of a species? Speeding up the rate of evolutionary rate of species. As A, lack of environmental changes, increase in mutations, C, decrease in migration, and D, increase in death rates. Something that can increase evolutionary rates of a species.
B, increase in mutation.
Correct. So if you have mutations, you create new, like new organisms can arise through mutations, like the way the coronavirus is mutating. Sometimes it's a decrease new varieties. So it speeds at evolutionary rate. Okay, mutation, correct. Okay, let's try another one here. Okay, I want you to make a guess on this one. We didn't touch on it, but try it. Page 248. 248. I'm going to test you on something first that we did. 32, then we'll do the other one that we didn't talk about. 32. So which of the following equations represent aerobic respiration? I remember I said you should know that offhand for respiration and photosynthesis backwards. So which one represents aerobic respiration?
My guess is A.
A, perfect. That's correct. So aerobic respiration, so good guess. Have C6H12O6 plus 6 oxygen. So it's a carbon, a glucose plus oxygen. So that's respiration, giving you carbon dioxide and water as waste products plus ATPs, energy, 38 ATPs. So that is respiration, aerobic respiration. So remember, aerobic, oxygen has to be somewhere to react to something. Okay, number 33, let's try that one. You can make a good guess on that. An overdose of muscle relaxant may cause a person to cease breathing because, so talk about muscle relaxant, something that relaxes the muscles. Because A, the diaphragm will be unable to contract. B, the lung muscles will cease to function. C, the muscles around the trachea will constrict. And then D, the alveoli will constrict. So what do muscle relaxants? Why would the person breathe? A, correct. The diaphragm will be unable to contract. Remember that the diaphragm is a big muscle. So it's a big muscle. You know, so muscle relaxant is going to relax the muscle in the diaphragm. It helps a person will have difficulty breathing. Okay, but let's try 34. 34 has to do with neurons, transmission in the neurons, nerve. Say a decrease in acetylcholine synthesis in humans would most likely lead to A, an increase in protein digestion, B, a decrease in nerve impulse transmission, C, an increase in muscular activity, and then D, a decrease in white blood cell activity. You are decreasing acetylcholine synthesis. Acetylcholine is released at the nerve ending, right? That's, you know, at the neuromuscular junctions.
So B.
So to decrease the nerve impulse transmission, correct. You know, because you release a nerve ending and then stimulate the muscles to contract. So if it's down, then you're going to have like reduce impulse. Correct. Okay, so sometimes you can make some intelligent guesses even if you don't know, you know. Okay, let's see this one here, page 250, 250. So that has to do with calculation of the heat, amount of heat. So I'll give you like one minute to try that. 46, 250, number 46. It says the specific heat of water is 1 calorie per gram per degree Celsius. How many calories of heat are needed to raise the temperature of 30 grams of water from 25 degrees Celsius to 75 degrees Celsius? So if you can try that in a minute or less. Calculate amount of heat. So remember the formula, Q is equal to MC times the change in temperature, T2 minus T1. Okay, did somebody get the answer?
Should I try?
Yeah.
C.
C is correct. That's correct. Okay. So you see, the mass is given. So Q is equal to MC times change in temperature, T2 minus T. So if you write this down, so this is equal to, you know, the mass is given as 30 times the specific heat capacity is 1 calorie per gram per Celsius. And then the change in temperature here, you have 25 minus 25. So you have 30 times 1 times, this is 50. So you have 1500 calories. Correct. C. That's correct. Good job. All right, so that's one heat calculation problem. Okay. Then look at page 251. Page 251. You have to do from the genetics problems. It says in a pea plant, tallness is dominant over shortness. Right? Tallness dominant over shortness. Now, if 25% of one generation of pea plants is short, what are the probable genotypes of the parents? So here you're working backwards. You want to find a genotype of the parents to get the percentage given to you. All right, so if you can do that in a minute, that will also be great. So here, work backwards from the answer and see which one gives you the percentages needed. So try that in a minute. Do elimination.
My guess is C.
Your guess is C. Okay, let's see if it's a good guess. So you have C, uppercase, so the Punnett square. So let's start with C. So this T, and then uppercase T, lowercase. So this will be T here, T, this and that, T, and this and that, T. So if I look at this, I have how many tall people here? It's a 1, 2, this is tall. They are all tall. They are all tall here. But we want 25% short. So it means that this it doesn't help us. So this one doesn't help us here. So C is out.
D.
B. Okay, let's see B.
D.
It's a what?
D.
Oh, B. Okay. So let's try
D.
B. D.
D. Yes.
Okay, let's try D. So D, you have uppercase, so T, lowercase, uppercase, lowercase. So this is T. This is T, uppercase. This is T, and this is T. Okay. So if you look at this one, we see that we have, this is tall, and then this one is short. This is short, right? So that's a short person. So this is 1 out of 4 short. So 1 out of 4 short. That means 25% short. So that's correct. So it means D is the correct answer. It gives us a percentage, 25% short and 75% tall. Okay, so that's how you use a Punnett square to work out these type of problems. Okay. All right, let's look at another. One more. Let's see. page 294. Page 294. Number 32. It says blood returning to the heart from the body travels through the vena cava and enters the A, right atrium, B, right ventricle, C, left atrium, and D, left ventricle.
Left ventricle.
So blood returning from the body travels through the vena cava. So talk about the vena cava.
Right at the A.
A, yeah, right atrium. Enters the right atrium. Correct, right atrium. Now what about 31? The enzymes found in the stomach and small intestine.
31, the answer is D.
Is D, correct. So lipases, they break down fats. Okay, lipases, correct. Lipases, okay. Page 296. Look at number 40. Although it is not possible to experiment with human beings to obtain genetic information, data on human genetics have been obtained from A, observing human eggs under a microscope, B, listing the genetic characteristics of individual humans, C, fertilizing human eggs in a glass dish, and D, making family trees that include information about certain diseases that related individuals have had.
D. The
D,
answer
correct.
is D.
Family tree. That's how you collect information about, some genetic conditions. Family tree, the tree diagram. Okay. Now look at number 39. We did that last week, I think, under the mechanics. See if you can remember 39. It's a calculation. Physics. An unbalanced force of 5 newtons causes an object to accelerate 2 meters per second squared. The mass of the object in kilograms is, so think about that if you can remember what we did last time.
I'm sorry, what number are we on?
Number 39, page 296. 296, 39.
39, right, Professor?
Yes,
I did the C.
you had C. Okay.
Yeah.
All right, that's correct. Okay. So remember the formula. We said force is equal to the mass times the acceleration. Mass times acceleration, that's how you calculate force. So the force is given to you, so 5 newtons is the force. The mass, we don't know. So let's use M, and then the acceleration is given. So it's 2 times M, 2M. If we divide by 2, this will cross out. So your M is 5/2, which is 2.5. Okay, 2.5 kilograms. Okay, so remember, force is mass times acceleration. Okay. Okay, let's try number 41. Number 41. The region of the human brain where speech function and conscious thought process are located is called A, cerebrum, B, cerebellum, C, medulla oblongata, and D, hypothalamus.
Is it B? Cerebrum.
A or B?
A.
A, correct, cerebrum. Okay, so that's like higher thought processes, thinking, and so on, cerebrum. Okay, correct. All right, so I believe now you've seen some. All right, there's one more genetic problem, which I just saw here. Page 297. 297, number 44. Number 44. It says a cross between a plant with red flowers and one bearing white flowers produces offspring, all of which have pink flowers. This is an example of A, incomplete dominance, B, Mendel's law of independent assortment, C, Mendel's law of segregation, and D, sex linkage.
B.
Try again. We spoke of like a red flower, white flower. If you cross them, they become pink. So there's something happening here. What do you think is happening?
B.
Well, actually the answer is A, incomplete dominance, right? So the red is not completely dominant over the white, so it's in between. So it becomes pink, right? So look at the notes. I gave that example under the incomplete dominance. Incomplete dominance. So it's like a in between the red and then the white, so pink. Okay. All right. So I think this will be a good point to end on this for now. So practice some more questions like this, based on what you've done so far. All right, so let's pause here and now let's go to our math so that we can catch up and do a lot in the time left. All right, so let's go to where we stopped last time, I think we stopped on page 108. Number 25, that's where we stopped. So we'll try and finish this and go to the next section of the test. Okay. So try number 25, 26, 27, 28, and 29, and then we'll discuss them. So this is what that means. It means 2/5 times 1/100. Okay. So I'll change this to this number here, fraction here, to a decimal, since the answer is in a decimal form. So if I do that with your calculator, or if you don't have the calculator, they have to divide 5 into 2 like this. 5 into that is 0, impossible. Into the decimal point, add 0 to this. So it becomes 4 and 20. So 0.4. So 0.4. And then times 1/100. Okay. If I don't want to make any error, I can put this over 1. Okay. Now, it's easy to divide by 100. Okay, so I'm not going to do much. So 0.4/100 is what I have so far if I multiply horizontally. If you divide by 100, you just have to move the decimal point 2 places to the left. So it means I have to move this 1, 2. So I have to add 1 0. So it's 0.004%. No. We make an error here. We made an error here. Look at it.
Yes, it's
The answer is 0.4%. That's what.
yes, it's D.
It's D, not the A. All right. But this is, if you look at this, we're not changing to decimal. We just want a percentage. Anything over 100 is a percent. So you have 0.4 out of 100, which is 0.4%. So we have to be very careful when you're working problems like this. You're dealing with percent out of 100. So whatever number you get out of 100 is the percent. Okay. So if you divide, that means you change to decimal, but that's not what you need here. So we have to stop like this. Okay. So if you see fraction as in decimal, it's easy to change to decimal and then work like that. Okay. Number 26 should also be fairly easy for us. They give you the formula and you have to evaluate the formula with the information given. So what do you get for that one?
12 milligram.
12 milligrams. Okay. All right. So you have 8. Y represents the child's age, and they say the child is 8, right? So you're going to write 8 over 8 plus 12, and then times the adult dose, which is 30. Okay. Work this out first. So 8 over 20 times 30. We can cross out zeros. You can put this over 1 if we don't make any errors. Then I can cross zeros out. Zeros is cross out. Then 2 into itself is 1. 2 into 8 is 4. So I have 4 times 3 left, which is 12. That's correct. Milligrams. Or if you're using a calculator, I can just multiply 8 by 30 and divide by 20 using a calculator. Okay, great. Okay, 27. So which of these problems is the value of 2 over 3 change by the calculation? Okay, so A, would that change the calculation? You have 2 over 3 plus 0. This is 2 over 3. Anything plus 0 is still the same number, right? Now for B, so that's A. For B, so this cannot be the answer. And then B, I don't know which book. Sometimes some books have greater than. It should be 2 over 3, 2 over 3 over 1. It should look like this. You know, in some older books, they have like less than, instead of the slash, it's like a less than here, you know. So if it's like that, your book fix it. It should be slash like that. Okay. So any number divided by 1, it gives the same number. So this one will remain the same number. What about C? 2 over 3 times 3 over 3. 3 over 3 is 1. Any number times 1 is the same number. So this remains the same. So it means the answer should be D. So this is out, and then D. 2 over 3 divided by 2 over 3. Now this is going to change because when you are dividing, you keep the first fraction and then times the reciprocal of the second one. Take reciprocal of the second and multiply. So if you work this out, you're going to have 1 as your answer. Because this will cross out, 1. So it changes. So the answer is the D. It changes the calculation. It's changed by the calculation. D. Okay. Right. Right. So for 28, it said the two short sides of a right triangle are 12 inches and 9 inches. How long is the third side? You know, so I put it in the diagram form. Right triangle, one side is 9, the other side is 12. What's the longest side? How long is the third side? The two short sides long. So what comes into your mind for a problem like this? Anybody, something should come to your mind. Right triangle.
Pythagorean.
Yeah, Pythagorean theorem. So you have to remember the Pythagorean theorem. Okay. So let's review that here. So it says that if you have a right angle triangle, if this side is A, the two short sides A and B, let's call it A and B. And then the third long side, the longest side, hypotenuse, let's call it C. Now he's found out that if you square the first short side, plus the square of the second short side. It gives you, and they add the 2, you get a square of the long side. So a squared plus b squared is equal to c squared. This is the Pythagorean theorem. Okay. So you can apply the same knowledge here. We have 2 short sides. So this is your a and b. So it's 9 squared plus the other short side 12 squared should be equal to the other long side squared. Okay. And then you solve this. So it's 81 plus 144 is equal to x squared. Okay, if I add this, this is 5, 255, 225 is equal to x squared. Yeah. To get x by itself, you take the square root. Take square root. So we're going to take square root of the left side and square root of the right side to get x. So square root of x squared will be x, and then square root of 225, and then that will give you 15. Okay, so 15. So that is a Pythagorean theorem that you have to use here. Okay. All right. Did anybody get to try 29? You have to think a little bit deeper for 29.
I didn't do anything on this page, Professor.
Not yet?
No.
Okay. So try a little bit. All right, so look at 29. It says, suppose that 3/4 quarts of blood have been donated for blood transfusion and 2/3 pints have been used. How many pints are left? How many pints are left? So the amount donated is 3/3 quarts of blood. Okay. And then amount used, we have 2/3 pints. So I've donated and used. Okay. Now the first thing, if you look at this, the units are different. So you can't work with different units. You have to have the same units. So that will tell you that you have to do some conversion somewhere. Okay, you have to do conversion. So you have to think about the relationship between pints and quarts. So we know that if you have 2 pints, it's equal to 1 quart. That's the relationship, 2 to 1. You have to remember this conversion. 2 pints is 1 quart. Okay, so we can change the donation amount to pints. So let's do that here. So if we change this to pints, it means that we have to multiply by 2. So it's 2 times 3/3 to change to pints. So this way you have to remember how to multiply fractions, multiple fractions. So to multiply the fractions out, we need to change to improper fraction. Improper fraction. So I'll change this to improper fraction. So I'll have 2 times 4 times 3 is 12 plus 3. So that's 15 divided by the 4. So that's improper fraction, 15/4. Okay. So if I multiply this out, what would I get? I'll end up with 30/4. Right, 30/4. That's which is the same as 30/4. I can reduce to lowest terms. This 1, this will be 1, this will be 2, So 15/2 as the pints. In other words, we have what we have here is 7.5 pints of blood. 7.5 pints. Okay. Now you used 2/3 pints of blood. So say how much is left? How much left? Yeah, so how much left? That means that you have to subtract, right? You're going to subtract something. So the total that we have is 7.5 minus what you used. You used 2/3. Okay. And since you're subtracting, we can use, you need to have LCD here. So 7, this is 4. This will be 2 minus 2/3. You're subtracting this, right? So LCD is 4. So I write this with LCD of 4. This times 2 to get that. Okay. Now, if I subtract this, I realize it's going to be a negative number, but I can borrow from the 7. So if I borrow 1 from the 7, I'll have 6 whole numbers left. But the one that I borrowed from the 7 is always in terms of the denominator. So that one is 4/4. That is the one that you borrowed, 4/4. So I add a 4 to the 2, so that makes it 6/4 over here. Okay. Minus 2/3. If we subtract now, 6 minus that is minus 2 is 4, and 6 minus 3 is 3/4. So that will be your answer, 4/3. 4/3. Now some people will change it to improper fraction and subtract, and it'll change back to a mixed fraction. Whichever we want to do it, that's fine. But I think borrowing makes you move a little faster. Any question on 29? All right, so you should be able to do a problem like this now. Okay, so the answer here should be C. That is C. Okay, so let's try page 109, number 30 to 35. 30 to 35.
Professor, same page, right? You said 35.
Page 109.
109, yeah, same.
Yeah,
Okay.
the next page. Number, like all the questions on that page.
Oh, yeah, next page, yeah, right. Thank
Yeah.
you.
So what do you get for 30?
I got B.
So 30, you can work backwards from the answer. Anybody for 30?
B.
B. Correct. Right, because S cube, so it means the number multiplied by itself 3 times. You know, so it's 6. 6 times 6 times 6. So X is equal to 6. That's correct. 31 percentage problem. Okay. So 31, you can always think of the percent proportion. Let me highlight the percent proportion a little bit here. Percent proportion. So that can help in a lot of problems. So have part over whole, or sometimes we use that the word the base, is equal to the percent out of 100. The whole always goes to 100. Okay, this is known as the percent proportion. It can help you in a lot of percent problems. How do you identify the part? To identify the part, always think of the word is, and then the whole or the base, think of the word of. Okay, like if I say John is a student of HCCC. Right. So the word is means part. So John is, that's a part. Anything associated with the is a part of something. So looking at the base or the whole of that, or the whole, base and whole. Okay. If you identify this in a question, you never go wrong. Is portion, of is a base. Okay. So what percent of 180? So of 180. So it means that this is the base that you're looking at, or the whole. And it is. So this here, associated with is, this is your part. Okay. So if you want to use a percent proportion, then you set up the part over whole, which is the is. Some people use the word is and of, like is and the of, like you know, that can help you identify them, is and of. So is, so that means 225 is the part of the base, which is 180, of, is over of. And this is equal to percent. I don't know the percent, so I use X over the whole, which is 100. The 100 is always a percent out of 100, the percentage. So this will be a percent proportion. And if you solve this, you have your answer. It's going to help you work out for all your percentage problems. You know. So now to solve for X, you do the cross product. So this times that, 180 X is equal to 225 times 100. And I can use your calculator to solve this. So divide by 180, divide by 180. So this will cross out and then have your X, and then you get your answer. You know. If you want to reduce to lowest terms first, you can also do it, but you're allowed to use a calculator. So just have to use it. Okay. So if you do that, I believe you should get 125 for it. 125%. So this percent proportion will become handy if you're not too good at percentages, use this one. It's going to help you out a lot. Percent proportion. They ask a lot of percentage problems, so you have to get used to them. And then number 32. 32 is also like a percent. It's a percentage problem. It's a percent problem. So 380 of 1900 wanted pure water. So let me read the question. It says, in a survey of community needs, 380 of 1900 respondents said they wanted purer water, purer drinking water. What percentage did not request purer drinking water? So a keyword did not. Okay, so always circle, underline the keywords that did not. So how can we approach this problem? Anybody wants to talk us through how to approach this?
Yes, Professor.
Yes, please go ahead.
First of all, you divide the 190 over the 38, which will give you 5.
Mm-hmm.
With the 5, since it's in percentage, you divide it by 100. So 100 divided by 5, which will give you 20.
Mm-hmm.
5 will go into itself 1, 5 will go into 120.
Okay.
So you have 20%.
Okay. So you have 20%. So we did not request, but the question said did not request.
What percentage did not request purer drinking water? 20%.
But I said did not request.
Yes.
So you have to subtract from 100, right?
Yes.
Did not. Okay. Okay.
Did not, yeah.
Yeah. Mm-hmm. All right. Great. So what she's saying is that there are 2 ways to approach this problem. 2 ways. So the one way that she approaches is to find the percentage. So let me call it method 1. What she did. So she changed this, she found what percent the 380 will be out of the 1900. All right. So you can do 380 as a part over the whole. Okay. And then she changed to percentage, X over 100, if you're using a percent proportion for those who want to use a percent proportion. Okay. If I'm doing this, I'll just multiply this by 100. I'll get the answer. All right. But this is a percent proportion. So this takes you to 1900. I'm writing everything for you to see. And then cross product 100 times 380 divide. So if you work this out, that's where she had 20%. 20%. So that is those who wanted. So wanted, what you did here is a wanted. So my question said did not. So since it did not, if 20% wanted, the total percentage is 100. So 100% is the total minus those who wanted. So that will give us 80% did not want. %80 did not want. Okay. So that's method 1. The second way of doing this problem, method 2. I can find how many did not, the number itself, right? Did not, those who wanted first. So those who want the number that wanted, I can find the number by subtracting. So that would be 1900 minus 380. I can subtract this to get those who did, no, this did not want. Let me change this here. The number that did not want. Number that did not want. That's what we are doing for this part here, by subtracting those who wanted. So if I subtract this, then that gives me 0, 1900, 380. So that's 0. That is 2 here. I have 18. So that's 5. 1520 are those who did not want. Then I'll change this to percentage. So the percentage that wanted, that did not want, percentage that did not want. So I take this, so 1520 divide by the whole, so 15, 20, 20 out of 1900. Then I can multiply by 100 to get a percentage. If you multiply by 100, then give you the percentage. So if you do that, you get 80% right away. So that's the second approach. If you're not too sure of this, use the percent proportion and do 1520 over 1900 is equal to X over 100. That's what all this means. What I did here, that's what it means. Cross products. You know. In other words, when you have a fraction, if you multiply by 100, you get a percentage. That's what that means. Yeah. So 2 ways to do the same problem. Subtract first, get those who did not want, and find a percentage. Or find a percentage that wanted and subtract 100%. You get a percent that did not want. Great. All right. So number 33. That one should fairly straightforward. 3/4 of 1 over 2 plus 1/4. Follow the order of operation for this question. Of means multiplication. So what do we get here? Three over 4 times, and then you have to find LCD, right? You need LCD. So the LCD here will be 4. So this is times 2 to get 4. So this times 2. So this should be 2 here. This is 1. So you have 3 over 4 times 1 plus 1, 2 plus 1 is 3 over the LCD, which is 4. So you're multiplying 3/4 by 3/4. And that gives us 9 over 16. So you should get 9 over 16, which is C. 9 over 16. So remember when you are adding fractions, LCD, very important, LCD. Okay. Number 34. Number 34, I'm going to talk a little bit about it so that when you see it again, you know what to do. So that has to do with what called similar objects. So here we are looking at similar triangles or rectangles. In this case, you have rectangles. So rectangle, similar rectangles. Okay. When you have similar rectangles, and one of them is an enlargement of the other, or a reduction of the other. Similar triangle means the ratio of the corresponding sides are the same. So let's say that this is A, and this length here is B. I have a bigger one. They look similar. This side here is C, and let's call the width here D. Because they are similar rectangles, the corresponding sides are related by the same fixed ratio. In other words, if I divide A, the length by the width, the ratio of the length to the ratio of the width, or the first one should be the same as the other one, length 2 over width 2. This is a proportion that you can use to solve these type of problems. Okay. In other words, ratio of A to B should be the same as C to D. You just have to be consistent. ratio of this to that should be equal to this to that. Same ratio. And it should be equal. So for this question, 34, if I draw the diagram, this is how it looks like. Two rectangles are similar. One rectangle has a length of 12 and a width of 8. So it's 12 by 8 centimeters. Then there's another rectangle. They said the other rectangle has a length of 15. What is the width? I don't know. So I use X to represent the width here. Since they are similar, they are related by the same fixed ratio. So the ratio of the width to the length, or the length to width, 12 to the 8, should be the same as the ratio of the length here, which is 15, to the width here, which is X. So this is a proportion that you can use to solve. So a lot of proportion in this type of problems on the test, a lot of proportion approach. Then you do your cross product. 12 times X, 12X is equal to 15 by 8, and then divide by 12. You can use a calculator to do this, or you can reduce to lowest terms if you want to.
B.
You got the answer already?
B as in boy.
B as in boy, okay. So you got B, all right. Okay, so this is 2, 4 here is 2, 4 here is 3, 3 here is 1, 3 here is 5, so 10 centimeters, correct. Yeah. Use your calculator, it's faster, since you're allowed to use it. I like to cross things out easily. Okay. All right, so you should be able to do similar triangles. It's going to come up in questions over and over again. Ratio of the corresponding sides are the same. Okay, look at 35. 35. It says a certain drug constitutes 2% of a solution. If there are 30 ml of the drug available, how many milliliters of solution can be made? All right. So again, this question, proportion. A certain drug constitutes 2% of a solution. So write down 2% of the solution means 2 of the drug in 100 of solution, 2%. So 2 out of 100. That's what it's given to you. The 2 constitute a drug. So it means have 2 of the drug here in 100 of solution. That's what that means. So we set out the same ratio here, have the drug and solution. Match them. So we say if there are 30 ml of the drug available, 30 ml of the drug. So 30 goes to the drug, goes to the 2. 2% is a drug. So it's 30 of that. Now I say how many milliliters of the solution? We don't know the solution. So this is X, unknown. You have a proportion that you can easily solve. 2 over 100 is equal to 30 over X. And cross product. 2 times X, 2X, then 30 times 100, and then divide by 2, divide by 2, okay? So 2 here is 15, 15 times that, so 1500. So here 15, so 1500 ml. Okay. So that would be D for this question. Okay. So proportion, proportion, proportion. Don't forget proportion. Works out a lot of problems. Any questions so far on this one? Okay, so look at page 110. Page 110, number 30 to 40. Page 110. Number 36. It says A is equal to 1/2 H, B1 plus B2. And the question says find H. You want to find H. Okay. So a question like this, you want to isolate H. You want to get H by itself. H by itself. Okay. So what do you think I can do? First approach. Again, this comes down 2 ways. But one thing I would say is that when you have a fraction, always get rid of the fraction. It makes your life easier. Get rid of fractions. So to get rid of fractions, multiply by the reciprocal of the fraction, or use the LCD, this common denominator, for the fractions to get rid of them. Okay. So if I look at this, I have the denominator of 2. So I'm going to multiply both sides of the equation by 2. Okay, so I've said 2 times A, and then I'll multiply the other side also by 2 to get rid of the fraction. So I'm going to use the red. So I multiply this by 2, and I multiply this side also by 2, like this. Or 2/1. We don't want to make any mistake. Reciprocal of the fraction that you have. So the 2 will cross out. So I have 2H, 2A is equal to H times B1 plus B2, like this. I want to get H by itself. So I ask myself, what is the H doing in this question? The H is multiplying, right? It's multiplication. It's multiplying the whole of B1 plus B2. So how do I undo the multiplication? I undo multiplication by dividing. So I'm going to divide by the whole of B1 plus B2. They are together, so divide once. So I divide the left side by B1 plus B2. So we have performed the inverse operation by dividing. Multiplication, inverse division. So I divide by the whole of this, they are together, that's why in parentheses. Whatever I do on the left side, I do the same to the right side. B1 plus B2. Okay, so this crosses out, the whole of this will cross out, be the same. So we end up with just H on the left side or the right side. So H is equal to the whole of this, which is 2A over B1 plus B2. Okay, so this becomes our answer. 2A plus B1 plus B2. So that will be A. A becomes your answer. So you should be able to isolate variables when they give you a formula. Yeah. If they are fractions, get rid of the fractions always. Okay. Any question on this particular one? Everybody's fine with that? Okay. All right, so look at 37. Number 37 needs a little bit care for 37. Ratio and proportion. A medication has 7 parts of drug A to 7 parts of drug B. If there are 420 ml of the medication, how many ml of drug A are there? Okay. So sometimes summarize in diagram form. So we have drug A and B. So A and B. This is 3 parts here. And this is 7 parts of B. Now I said if there are 420 ml of the medication, so the whole medication, all this is the total of 420 ml. The whole of this. I said how many ml of the drug A are there? Okay. So they give you some parts and they give the total. So you need to be careful in this question. Okay. So what you have to ask yourself is, what is the total number of parts that you are dealing with? The whole thing, we are dealing with 10 parts. we deal a total of 10 parts. Because 7 and 3 parts. So the total of 10 parts. That's what you're dealing with. So this total part goes with the 420. It matches that. This matches, that's the total part you're dealing with. So if I want to find what part A forms, then I have to use, you have 3 parts of A, out of a total of 10 parts. That is a fraction that A forms. So this will be the same as, I know the volume, the total part is 420. So 420 goes here. That's the total part, right? I want to find the part of A. So this is X unknown, X parts of A. So this is a proportion that you have to set up to solve. Okay, so this is X parts of A. And this is total parts. Or total volume. They have to match like this. Then you can do your cross products. So 10x is equal to 420 times 3. Then divide by 10, divide by 10. So your x will be equal to, and this will cross out. Then zeros can cross out. They have 3 times 42. So 3 times that's 6, and there's this 126 ml. So when I give you total volume, deal with the total parts. You got to deal with the total parts. Okay, like that. Any questions on this particular question? And so far, so good. Okay, all right, 38 then. 38 is simplifying. Simplification. Okay. Now, for you to simplify problems like this, you have to first factor out. If there are common factors in any problem, always factor out. So we need to factor this problem out, right? I see A here, I see A there. So I'm going to factor A out. So if I factor A out, I have A out, parentheses. Then A into A squared, this will be A, minus, divide this into that will be B. All over, common factor here is B. B out, so it's B minus, this will be A. Okay. Now, before we can cross anything out, they have to look alike. So I have subtraction. How do I make this look alike? This way you have to think about this. If I write 3 minus 2, sorry, yeah, 3 minus 2, right? This is the same as writing negative of 2 minus 3, right? If I switch positions in subtraction, I have to take the opposite of the answer to get the same answer, you know, because you don't have commutative property of multiplication. 1 plus 2 is the same as 2 plus 1, but it doesn't work out in subtraction like that. If I switch positions, I have to take the opposite. So negative of the answer, okay? So if I have A minus B, if I want them to look at, and B minus A, if I want them to look alike, they have to change the position of one of them. Change the position, so the switch A with a B, and then take opposite. So let's do that. So what I'm going to do, I'm going to switch A and B. So this will be equal to, I'm going to switch the position of A and B, so it becomes B minus A. Since I've done that, I have to take the negative of the whole answer. So I put negative of what I have here, negative of the whole thing. and then I can divide by B minus A. So now they look alike. Since they look alike, I can now cross out the common factors. And then you end up with negative A over B as your answer. Okay. So the key is factor out common factors first before you can cross out. If there's anything that you have to, if it's subtraction, you have to flip positions, you take the opposite, negative of whatever you flipped. So that is how you work out problems like this. Common factor first. Okay. Everybody's okay with that? Okay, so 39, 39, that one too, you have to think a little bit through that one. A rectangular room A has an area of 400 square feet. What is the area of a similar rectangular room, the size of which are all half as long as room A? Okay. So a problem like this, you can think in terms of diagram. I like using diagrams a lot. Summarize the question in diagram form. So you have two rectangular rooms. They look similar. I mean, there's a quick way of doing this too, but you have to know this before this way to understand it. So the first one, area of 400. So if you're rectangular, let's call it the length here is a length and width. So the area, we know that area of a rectangle is equal to length times width, right? So for the first one, they gave us the area here as, let's call it A1. Let's call the area here A1. So A1, area A1 is 400. So 400 is equal to the length times the width. I don't know the length and width, so I keep it like this. So this area 1, let's call it area 1. Then when we come here, area 2, it says that this is half as long as the other one. So half as long. So let me make it a little smaller. Half as long. So the width here is half and the length here is also half of that. Okay. So what will be the area? Area 2. So the area here again will be length times the width. So this is going to 1/2 the length times 1/2 the width. So that means that the area that I have here, area 2, is equal to 1/4 times the length times the width. This is what I have so far for the second area. But let's go back. What do you see here? What do you notice? The length LW here is the same as what you have here. Right? Same thing here. And what does that represent? This L times W is the 400 here. The first area, because it's the area of the first one, right? The area of the first one. So now I can put the 400 here, substitute there because it's the same. So the area that I need is 1/4 times 400. So that's 400. And when you do that, you end up with 100. Okay. So it's 100 square feet. That's the area that you want here. 100 square feet. Then your answer will be C. So you have to think like this, draw diagrams to help you out easily. Any question on this 39? So I believe everybody's fine with that one. Okay. All right, so number 40. Number 40 says, combined with 480 ml of water are 120 ml of alcohol. The percentage of alcohol in the resulting solution is, so again, this is a percentage problem. Okay. So combined with 480 ml of water is alcohol, 120 alcohol. So I can draw a diagram quickly to illustrate. You have 480 ml, this is water. And then here you have 120 ml alcohol. Okay. So what percentage of alcohol is the resulting solution? So you need to find a total volume, right? A total volume. So the total volume here, you have a total of 600 ml, the whole thing, total. So to find the percentage of alcohol, we have 120, that's the alcohol, out of a total of 600 of the solution. Okay, so that's a percentage, the fraction that it forms. 600 ml of the solution. Okay, so if you have this to change the percentage, we can multiply by 100. If you multiply 100, you get a percentage. Because that's a fraction. Multiply by 100, you get a percentage. Okay. And then if you cross out, zeros will cross out. 6 here is 1. 6 into 120 is 20. So this forms 20% alcohol. 20% alcohol. Okay. So that's how you play around with the percentage, proportion, and all those little things. So please get used to proportion. Very important to get used to proportion. Any question on what I've done so far? Any questions? Okay. So if you look at page 111, now the problems on that page should be easy for you. It should be easy for you. We'll be rounding up shortly. Okay. So that should be easy for you, page 111. Most of the things go repetition again. We're going to repeat. So yeah, the math doesn't change that much. You just have to get used to fractions, proportions, those things. Like number 1, it should be easy for you. We've seen 1 already. So I don't think we should waste time on number 1. You should be able to do that easily. Okay. 23 beats in 15 seconds. How many in 1 minute? So that should be easy for you to do. All right, we did something like that. If you look at number two, that one should also be able to do it. They gave you 3y is equal to 2x plus 8. And then they tell you that x is equal to 20. You want to find y? Question mark. So that one, again, you should be able to do it. You substitute the x for 20, and then you solve for y. Okay, you're replacing. So that will be 2x. So you're going to put that here. X is 20, and then you can solve. 3y is equal to 2 times that plus that. Okay, work this out. 40 plus 8 is equal to 3y. So combine this 48 is equal to 3y. Divide both sides by 3. And then you get your 16 for that. Okay, so that shouldn't take you too long to do that one. Number 3, you should be able to do it, number 3 yourself. Multiplying decimals. Okay, you should be able to do that. Number 4, fraction. You add in fractions. So if you're adding fractions, remember we said you need LCD for fractions. So number 4 is good to use to review your fractions. You can use that to review fractions. Adding fractions, the numerators are not the same. So you need LCD. So do 3 LCD, 4 and 5, least common denominator is 20. So write each of them with LCD of 20. Ask yourself what happens to the 4 to get 20. This is times 5. So if this is times 5, you multiply this also by 5. So that gives you 15 here. How do I get 20 here from 5? It's times 4. Whatever you do a denominator, do the same thing to the numerator. So this would be 16, all right? Same LCD, you can now add the numerators and then the whole numbers. So 3 plus 2 is 5. 15 plus 16 is 31 out of the LCD. But this is an improper fraction, 31 over 20. Improper fraction, so you have to change to a proper fraction. So if you change that to a proper fraction, you have to divide the 20 into the 31. 20 into 31, that's 1. That's 20 here, you have 11 left. So it's one whole number, 11 out of 20 parts left. Then you're going to add the 1, the whole number here, 1. You're going to add it to the 5. Right, so you have 5 plus 1. So you have 6 whole numbers. 6 whole numbers here with this fraction left. 11 out of 20. You know, because this is 11 out of 20. What you have here, okay? And then that would be your answer. So you can easily do this type of problems. Okay. And if you look at the last one, that one is volume. Volume of a cube. So volume of a cube, remember the volume of a cube is area times the height. So in other words, if we're dealing with a cube, it's length times the width times the height, okay, cube. So the volume of a cube is equal to the length times the width times the height. Okay. Length, width, height. And then you get a volume. So for this question, you have to multiply all the numbers. If you multiply all the numbers, then it gives you the volume. And that should be 1440 cm cube. Now, one thing about this is that you have to remember, sometimes you get the answer by the unit. The unit is important. The volume, it should be cube. If it's area, then it's squared. You know, so sometimes you have the answer, but then the unit, they can change it. Centimeters, then they write centimeter cube, the centimeter squared. If you're dealing with volume, remember it's cubic, cube. Whilst if area, you're squaring, so square. You know, you have to watch those things in problems too. Yeah. So I believe that would be a good point to stop for now. So practice on more of these type of problems. You're going to say there's going to be a lot of repetition in what you have done already. Okay. All right, any questions before we end for today?
No
I'm good.
question.
Okay.